to all my children. Hello, 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 hello. Am I audible? Am I visible? Hello, 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 hello. Yes. Oh my God. So many children were there waiting for me. Guys. Yes. So I think the audio is perfect. Can you guys give me 1 minute such that I can send the link in the group, and also I want to send one message in the group. Just let me do that and then it's all sorted. Perfect. Hello dear children, how are you all? Hello Hassan, hello Jia, good afternoon Mansi, good afternoon Purnima, Sanjeevani, Vinayak, okay, Gururaj, everyone. So I see a lot of children out there, and now listen to me very carefully. Okay, I want to be brutally honest over here. Sarvesh, hello. I'll be sending a message in the group. Read that particular message. It's super important. So those of you watching this, you can watch it, but those of who feels that I have not done anything in biology and who literally needs help, might not have too much amount of time also. So in that particular case, what I have done is I have typed a message for you. I'm pretty sure it is going to be helpful. Make sure you have a quick read of that, and I do not know how to copy it. Just a second. Okay, so you might feel like I have given a paragraph out there. Okay, so children, read that carefully and you will get the answer for all the questions that you have been asking me, that ma'am, if I have not done anything and things. Anyway, I'll brief it up. Listen to me carefully. The thing is that if you are someone who have not yet done anything in biology, there is a video on complete biology, where what has been done is that all 1 marker, 2 marker, 3 marker and 5 marker have been merged. You do not have to watch the MQP separately. Why? Because model question paper questions also have been included up there. The duration of that particular class is 10 hours. It was a recorded lecture, which is why there would not be any talking in between. It's a straight lecture which is there. You can increase the speed and watch it. If that is the case, please move out of this particular live. Go watch that if you have not studied or touched biology even once. Okay, now next thing. For the children who were there with me in the live yesterday and who have to complete the other chapters today, be here itself, because we are going to complete those chapters now itself. And as I said, I am brutally being honest over here, right? So when I say passing package, everyone knows there is no separate set of questions that I can invent, right? The same things were there that I told the previous day. So this was the video. If at all you have not watched these chapters, the name of the chapters goes like this. So basically, I have done sexual reproduction in flowering plants there, human reproduction there, reproductive health there, principles of inheritance, molecular basis of inheritance, evolution, human health and diseases, and microbes in human welfare, organisms and population. These chapters were done yesterday. Actually, these 2 chapters were also done, but I went literally very fast, which is why I know that there might be a lot of confusions, right? That is why I'll be repeating these chapters over here. Which one? Biodiversity and conservation, I'll repeat. Ecosystem also, I'll repeat. Biotechnology principle and processes, biotechnology and its application, I didn't do yesterday, I'll do it today. So what do you understand? If you want to do the other chapters that is being mentioned over here, which I've already done, you go back to this particular live, watch it. If you want to cover these chapters with me today, be here, watch it. Okay, or probably first watch these things and then when you have time, go back. Now I have given the timing also for each chapter. Sexual reproducing flowering plants, this is the timing that is there. Roughly 30 minutes was given for that particular chapter. The same goes for everything else. So molecular basis of inheritance, roughly 25 minutes. Evolution, roughly 40 minutes. I have covered in the previous live. Please watch that live. Now I am planning to cover these chapters here, and if I have time, I will be repeating these chapters as well. The live, I am thinking of ending it by 6:30 p.m. okay, or probably 6 p.m. such that I can take another live at 7:30 p.m. Directly, I'm going to take questions from the question bank for the MCQs for the chapter that gives lots of MCQs. For example, these chapters that you see over here, 2, 2, 2, 2 and 3 marker. Does that make sense to all of you? Is it fine? Hello Bhuvan, hello Sanjana Sanju, and hello Vittal. Yes ma'am. Okay. So I do not want to waste on your time. That's why I told this. Fine? Works. Perfect. Great. So let's start ahead with biotechnology principle and processes and biotechnology's application first. And the question number remains the same. So from that PDF you can refer. That is why I have not changed the question number. Hello everyone. Hitashi and Sangamesh. Okay. First question. Statement number 1. Plasmid DNA acts as vectors to transfer the piece of DNA attached to it. Statement number 2. Vector DNA act as plasmid to transfer the piece of DNA attached to it. And you have the options over here. Give me the correct answer for this. 48A. Mansi says 48A. Hello Malika. Okay. 7:30 there will be a class of MCQ. Yes. A. Bhuvan also says A. What about others? What about others? 48A, Jia, Hassan, everyone is saying A. A is what? Statement A is true, but statement 2 is false. Is that so? Listen to me carefully. Plasmid DNA act as vector. What is plasmid DNA? Everywhere in a bacteria, okay? In a bacteria, they will be having its own genetic material. Apart from that, there will be the extra chromosomal thing. Remember, these are the features of plasmid, super important. What is this? Extra chromosomal in nature. It is double-stranded in nature, double-stranded. And basically, they will be having origin of replication of themselves, so that they can replicate. And they do not code for any big proteins out there which is necessary for the organism to live. So what are we going to do? We are going to take this particular plasmid out, since they do not have much bigger functions. And basically what do we do is that we make a cut over here. Why? Because we want to put our gene of interest in between. Now when we put our gene of interest in between, what is going to happen? Basically, whenever the plasmid is going to replicate, this particular portion will also replicate. Now what do we do? With the help of this, we are going to put it in a host cell. Host cell, for example, for any other bacteria we will put it, or any other organism we will put it. What will happen? It will normally replicate out there. So basically, isn't it acting like a carrier? Baba, it is acting like a carrier over here, right? It is helping me transfer things, and these carriers over here are known as vectors. So definitely I can say that plasmid DNA act as vector to transfer the piece of DNA attached to it. Now the next statement is vector DNA act as plasmid. Listen to me very carefully. Vectors can be anything. It does not necessarily be a plasmid. Here plasmid is acting like a vector, but it can be any other thing. For example, sometimes a retrovirus can act like a vector. Is retrovirus a plasmid? No, it's a whole different thing. So I cannot say that all vectors are plasmid. I can say that plasmids are vector, but I cannot say all vectors are plasmid. So here it is said vector DNA acts as plasmid. That would be a wrong statement. So statement 1 is true, statement 2 is false. If at all you got it, please give me a thumbs up. Please give me a thumbs up. Mahantesh, ma'am, all the important questions, dear. Go join the WhatsApp group. I have given you the video link as well, the PDF link. Read the message. It is for you guys only. Yes, ma'am. Perfect. Okay, next question that we have is, in the given diagram of PBr322 vector, identify the restriction site A, B, C and D. So over here you have the restriction site A, B, C and D. You have to tell me, it's not basically restriction sites, they are just sites. You have to tell me what is the answer for this. HOTS questions are going to be covered here only, and in the yesterday's live also HOTS questions were being covered. Yes. Ma'am, I'm seeing your 2 marker, 3 marker, 5 marker. Can I score full? You should be able to, and also watch the 1 marker video just to be safe. And also, if you have time, the HOTS question that I've dealt in the previous lecture, that is yesterday, just read through it. Okay, anyways. Hello JJ, ma'am. Hello, Tara. Okay, so most of you are saying B, and I see C also. B and C. Why C? So most of you guys are saying B, which is why I would log B, and I'll tell you a way to remember plasmid, PBr322. This is very important. Okay, so listen to me carefully. You have the plasmid like this, right? Obviously, all the plasmids will be having origin of replication. This is where the DNA polymerase is going to attach. Listen to me carefully. Now, apart from it, it also has one more thing. What do you call this site as? ROP site. ROP stands for replication protein. So there is a protein that is made that is super important for the replication process to happen. So that protein has been coated by the ROP site. Now, this particular plasmid has 2 resistance gene. Resistant gene basically means that this particular gene gives us a superpower. Superpower of what? Superpower of basically sustaining in a medium that has an antibiotic. You know bacteria killed by antibiotic, right? But if at all they have ampicillin resistant genes, they will not be killed by ampicillin, which is an antibiotic. So this is a gene for ampicillin resistance. Over here they have another gene that is for tetracycline resistance. These both things are what? Antibiotics. Now comes to the point, that is basically the restriction enzymes. Listen, there are 8 restriction enzymes that have a site over here. The restriction enzymes, what do? They make a cut, and basically when they make a cut, we put a gene of interest out there. You call these sites as what? Cloning sites or restriction sites, and you have 8 of them. And how do you remember them? Remember, there were 2 guys, okay, they had one pista shake, okay, PST1. And then they also had vada pav, one vada pav, okay, PVU1. PVU1 is for vada pav. So pista 1, pista shake 1, and PVU1, vada pav 1. Done. Now after that, what? Basically, they were economic students, okay. They were economic students, Eco R1. And they went to a class. CLA1, CLA 1. And this class was Hindi class. This class was what? Hindi class. And the Hindi class, my dear children, you have to remember Hind 3, okay? Hind 3. Or probably you can remember 3 guys were there. They had one pista shake, one bada pav. They were ecology students. They went to the class of Hindi, okay? And yes, that's all. Now coming over here, you have 2 more sites. What are this? This is BAM H1. This is what? BAM H1. And this is SAL1. Okay, and now when they got into the class, my dear children, they bammed with what? Salman. They bammed with Salman. This is how you can remember it. I do not know. And then when they bammed with Salman, they all went out. They all came out over here. They thought that we should be having one more vada pav. So basically, PVU2. PVU2. This is the only way I can make you help you remember these things. So basically, ROP site. In the ROP site, you have PVU2. PVU1 was right over here, and everything is right in front of you guys. Okay, I know, okay, I know that is not the best way to remember things, but yes, I just wanted to help you guys. So I'll just, recollect once more. Listen to me carefully. Origin of replication, ROP site is there. Yes, ROP site is the protein that is super important for replication to happen. Here you have ampicillin resistant, here you have tetracycline resistant site. Done. Now what happens? You have pista shake 1, and then you have one vada pav. You went to the eco class, okay, and basically ecological students went to the Hindi class, and this was the 3rd hour of the class, and they bammed into Salman, and basically all of them had 2 vada pav. That's all, okay. Thus, so over here your question is what? A, B and C. What is A over here? If at all, now you look at this particular picture, this would be origin of replication, ORI site, okay. This is going to be tetracycline resistance. This is going to be what? Ampicillin resistance. So ORI site, ampicillin resistant, tetracycline resistance. And what is this particular thing? So always remember, when you have 2 Dundies like this, one is for CLA1, another one is for Hind 3, other one is for Hind 3. So D is going to be Hind 3. So your answer in that particular case is going to be B. It is going to be B. Okay, got it? Perfect. Yes, yes. Okay, now coming to one more important thing. Suppose, my dear children, I want to give you an important information about this, guys. I am saying that I am using BAM H1 for cutting the gene out there. If I use BAM H1 for cutting out over here, you need to understand that it will lose tetracycline resistance. Why? Because this was a gene for tetracycline resistance. In between that, I am making a cut and putting another gene over there. Do you think it will be tetracycline resistant anymore? No. This is known as insertional inactivation. You're inserting something, which is why other thing was getting inactivated out there. So they might ask you, what are the restriction enzymes that you will use for insertional inactivation of, or basically for inactivating tetracycline resistance and ampicillin resistance? So you should know these 2 you will use for inactivating tetracycline resistance, and these 2 you will use for inactivating what? Ampicillin resistance. This is super important. What is the thing that you will use for basically not allowing the replication protein to be made? It will be PVU2. So these sites are important in that sense also. And also one more very important thing, if at all you see BAM H1 is over here only. Do you have multiple BAM H1? No. You do not have multiple BAM H1. Why? Because if BAM H1 has a site over here and here, it will make 2 cuts. Do you think the plasmid will still survive there? No. So each cloning site will be having only 1 site per plasmid. Per plasmid, only 1 site will be there. But You can have multiple restriction enzymes. For example, this is another, none of them are getting repeated. So please understand the difference. You can have multiple restriction sites, but one enzyme will be having only one site. One enzyme will be having only one site. Done? Clear? If this is clear, just give me a thumbs up. Everyone who is watching this video, if it's clear, just give me a thumbs up. And ma'am, duration. Ma'am, duration 6 o'clock, 6:30 we will finish it off. Okay? Just by studying 3 model question papers, what can I get? I cannot tell you that. Why? Because we are not sure about it. Listen, who gave the guarantee that the same question will be repeated just like how it happened in chemistry? No one, right? So obviously you have to do it, but you should not just rely on it. Please solve some extra questions as well. Yeah, works. Perfect. Thank you guys for giving me the thumbs up. I think I can go ahead with the next question out there. Yes. Hi Zainab. Hello, hello, hello. Hello Abhi. Okay. Perfect. Now, when you talk about this chapter, that is biotechnology principle and processes, you know you get 2 MCQs you get from this chapter, and then you get 1 5-marker question from this particular chapter, right? Yes. So what do we have to do? So basically, we have to look out for the topics that have most probable chance for coming in. PABR 322 related things can come in. Tools and processes involved here also have more chances to come. So make sure you practice them. Here are the new questions that I have not solved anywhere until now. Those also would be discussed right over here itself. Okay? I hope that works. Let's go. So first question that you have over here is, write any 4 tools used in recombinant DNA technology. Guys, come on. Any 4 tools that you are going to use in recombinant DNA technology. If at all you want to make a new combination of DNA out there, what are the things that you're going to use? What are the tools that you're going to use? Any idea? Everyone, come on. Yasin, hello, hello. Pranav, Kamble, and Law of Traction. Oh my God, so many children are there. Bunny, there will be definitely free content as well. Pranav, I read your name. Yes, guys. Micro-injection. Okay. Hassan said micro-injection, restriction enzymes. One person said, okay, Rashmi says restriction enzyme and DNA ligases. Yasin also said DNA ligases. Yes. So listen to me carefully. First thing that we need over here is a restriction enzyme. Why? Because restriction enzyme is the one that makes the cut. Okay. Now, after making the cut, you need someone to join as well, right? Who is the person? Basically, suppose I have my gene of interest over here and this is the plasmid right over here. I need to join the stuff over here. Who does that? With the help of DNA ligase. What does the DNA ligase do? Will be making what? The sugar phosphodiester bond out there. Okay. They will be making the sugar phosphodiester bond out there. Super, super, super important. Now, apart from that, what do you need? You need vectors. Okay, you need vectors. For example, it can be plasmids, it can be viruses. Okay, this is basically what? Viruses. Anything that will be carrying DNA into the host. Now, vectorless methods are also there where you use microinjection, but those are methods that you have. Okay, those are actually methods. So if at all you're writing that, you have to write it at the end. Usually write the name of the vectors, the normal vectors out there, plasmid, viruses, retroviruses basically. When you write viruses, right, retroviruses and things like that. Finally, what do you need? You need competent host cells also. Arey Baba, you need to put it somewhere all na. Obviously, you need to put it somewhere. That is going to be what? Host cell. That is going to be a host cell, and that it can be an E. coli or any other organism out there where the recombinant DNA is going to replicate. Please remember, these are the 4 tools out there. Okay, there are the 4 tools out there. Obviously, you can go ahead with the methods as well, but at least write this. And yes, someone said bioreactors. Yes, 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 we need bioreactors also. For what? Again, this comes at the end. Do not write all this thing in the starting point. Okay, bioreactor is for what? Basically for amplification of the host cells out there, or basically the cells out there. Hello, Ritika. Okay, got it? Is this clear? Is the part A clear to everyone out there? Part A clear to everyone? Give me the thumbs up. Give me the thumbs up if at all part A is clear. Okay, fine, perfect, thank you. Mention any 2 methods of introducing alien DNA into the host cell. What are the 2 methods of introducing alien DNA into the host cell? Any idea, guys? How to score full? My dear child, go read the WhatsApp message. I have given you everything. Biolistic, Rashmi says biolistic and micro injection. Okay. Nayas, okay. What about others, my dear children? What are the methods that you have? Guys, I have dropped my pen again. Okay. Mansi says micro-injection. Okay. Micro-injection is what everyone is saying. Because that is easy to remember, right? So listen to me. There are vector methods where you use a vector to transfer things out there. Yeah, Akshita says gene gun, perfect. And there are methods where you do not need a vector out there. You call these vector-less methods. So what do you have? You have micro-injection. Okay, micro-injection is there. Now, do you guys need an injection? Well, obviously no, right? But in the case of an animal cell, my dear children, in the case of an animal cell, we actually do it. For what? For transferring the recombinant DNA. Suppose we have the recombinant DNA and this is our nucleus and we have to transfer it over here. What do? We take the help of a micropipette out there, or basically something that is sort of like that, and we put our recombinant DNA over here, and that is how we are going to inject it. It is very easy over here. Why? Because it does not have a cell wall. You're talking about an animal cell. It does not have a cell wall. That is why you take micro-injection method. Now, what about a plant cell? Can you go do the injection? If at all you do that, that particular nerve will be broken off. Why? Because this guy, the plant cell which is there, it has a cell wall, macha. Okay, it has what? It has a cell wall. So over here, this is a cell wall, and this will be the cell membrane, and you would be having the nucleus and the genetic material and everything out there, right? So in this case, I am going to use a gun. Instead of using injection, I am going to use a gun. What do you call this gun as? Gene gun. And this is something that everyone remembers. But Papa guy, they forget the other name of gene gun method. It is also known as biolistics. It is also known as biolistic method. Do not forget it. It can come. Okay, biolistic method. Okay, yes, someone is saying gold and tungsten. Yes, Hassan, gold and tungsten. So basically what do you do over here is that you take the gun and you have the particles made up of gold and tungsten. You wrap your DNA around the gold and tungsten and then you shoot. And then finally what happens? The genetic material will be right inside the nucleus. Please do not forget, it is the particles of gold and tungsten that you use. This is for what? This is for plants and this is for animals. This is for animals. Now, is there a separate method for in the case of bacteria? Is there a separate? It is only for the plant's DNA. The legend. This one is only for the plant's one, and this one, yes. Okay, guys. Now, one more question that I have for you is that, ma'am, where is disarmed pathogen? Disarmed pathogen, that is a vector. Listen, I'll tell you the basic difference. Vectorless method means you are not using any vector to carry out things. Are you using any separate plasmid or retrovirus over here? No, Baba. You're directly doing the job, right? That is why you call this vectorless methods. Fine. This is the one thing that you have over here. Now, one more thing that is there is what is the case about bacteria. In bacteria, what is going to be happening, my dear children? It's simple. It's simple, okay? In the case of bacteria, the thing is simple. Suppose you have the bacteria right up over here. Okay, and you have the recombinant DNA right up over here. The first thing that you will do to make the bacterial cells competent, the term competent comes and sometimes we think, oh my God, what is competent? Competent means the ability to take up something. So I want to make this cell able to take up the recombinant DNA. So that is what I mean by being competent. So how do you make the bacterial cell competent to take up the recombinant DNA? It comes in these methods only. Okay, so what do you do? First of all, you are going to treat the whole thing. You're going to put it in a medium that is containing calcium chloride. Why do you do that? Because macha, this calcium which is there, it is a divalent cation. It is a divalent cation. That means a bit about chemistry over here. Divalent cation means it has 2+ ions over here. Okay, done. What does this do? What is the use of treating with a divalent cation? Divalent cation is going to make everything neutral. What, ma'am? Actually, yes. But what do you mean by neutralizing things? Listen, our DNA which is there, it is negatively charged. Our plasma membrane also have some charges. Basically, if at all they have charges like this, it will not allow the entry of a DNA inside the cells out there. So what do? We have to make sure that all charges which is there in the DNA, okay, on the DNA and also on the plasma membrane, it is gone away. So that function of neutralizing all the charges out there is done by what? This particular calcium. This is one function. Second function, Second function is there, that is it creates temporary pores. The calcium which is there, it makes what do you say, it makes the things more permeable. Okay, it makes the membrane a bit more permeable, not exactly permeable, but a bit permeable. So I'll also write it makes it a bit permeable. Do not go and write entirely permeable. Okay, it makes it bit permeable. Now talking about the next thing that is up there, that is happening over here, is that first thing we treated with calcium chloride and all those things happened. Now we are going to put it in ice. Now we are going to put it inside ice. Now you might feel that I am some psycho out there. Listen to me carefully. So why do we put it in the ice? For some reason. For some reason. Okay, we are going to change up the temperature out there, and this temperature change is going to do something. So we put it in the ice, and then, my dear children, suddenly we are going to change this thing. We are going to give a heat shock. Heat shock. So at 42 degrees Celsius, we keep it for some time. So for some seconds out there. Why? Because this temperature change, my dear children, will be creating some pores. Already we made it a bit permeable. Now we have pores also. Some transient temporary pores are being made up over here. Why now? Because you want the DNA to get inside. By this particular thing, what happens? Your DNA gets inside. But now the DNA will go outside also. You need to prevent that. How do you do that? Again, you put it back on the ice. When you put it back on the ice, all these guys which are there, they will be closing up, and finally the recombinant DNA is inside the cell. This usually happens where? This usually happens in the case of a bacteria. Okay, so micro injection for animals. In the case of plants, you have gene gun and biolistic, and over here you have what, my dear children? You have the method of making things competent using CaCl2. Done. Is this particular thing clear to you? These are all vector-less methods. Competent means to be able to take up things. So how do you make the cell competent? By doing this. Okay, this is for bacteria. Ma'am, duration of this particular life, this life is going to end by 6:30 is what I believe. If I can end it fast, I'll end it fast also. Because for the next slide which is there at 7:30, I am going to solve the question bank MCQ, so even I have to go through it once. Okay? Ma'am, can you repeat what do you to read to score full? Okay, so first of all, listen to me very carefully. I can help you to get the best. Okay, I cannot guarantee you that you will get full on full. Can anyone guarantee? If at all anyone is guaranteeing, they will have the paper. So listen to me. But I can tell you to give your best shot. There is a video that I did previously on complete biology. It is a video that is like merged video. So there were 3 things that were done. All 1 marker, 2, 3 marker and 5 marker. These 3 of them were merged together chapter wise and were given to you. The name of that particular video is known as Complete Biology. In that you have the MQP questions. In that you have the question bank questions also. That is super important for you. Now there are some questions which are of bit more higher level. You watch that video, that is for 10 hours. So if you're someone who feels that I am least confident biology, I need help, I need terrible help, you go away, forget about every life that is happening up over here, take that particular video and start studying from it. That would be more than enough. Now, if at all you are someone who feels confident that I know the basic of things, I need to solve some extra questions just to be safe. I might be asked these questions in my examination. So yesterday I did a live where per chapter I did certain questions out there according to the MQP. I did not repeat the MQP questions. I solved some other questions related to the concepts which were there. You at least go through that concepts. Okay, if you cannot watch the video entirely, read the PDF. Everything is shared to you in the WhatsApp channel. Go through that. And I'll be not repeating this again. Why? Because it will waste a lot of your time. Yeah, and some chapters were left over here, which is what I'm going to deal in this particular live. Okay, hello, law of attraction. Okay, ma'am, what is your name? My name is Jasmine. You can call me JJ, ma'am. Okay, perfect. So this particular question is also clear. Now see question. Name the stains used to visualize DNA fragments in gel electrophoresis. So what is the thing that we use to view the DNA fragments in gel electrophoresis? Any idea? Any idea? Ethidium bromide. Yes, exactly. Someone said before also. So yes, Rehan and Rashmi and Hassan, everyone. Sanjeevani, Mansi, yes. What do? We have ethidium bromide. Please do not forget it. Ethidium bromide. What color does it give? Any idea? What color does it give? It gives a orangish red color. Okay, orangish red, sometimes it is written as brownish red. Just go through your NCERT regarding that, yeah. And also you need UV light to view, okay, you need a UV light to view under it, otherwise it will not be visible. Please have that thing also clear in your head. Okay, bright orange, exactly, bright orangish red. Okay, done. Now I hope this particular part is clear. I have written the answers carefully over here. So how does the ethidium bromide work? It gets into our DNA, okay, it gets intercalated, and that is how it is visible to us. Fine. Now, next thing that we have is a question related to give reasons for the following, origin of replication and things like that. But before I go ahead with that, children, I want to add up one more thing. So everyone got the vectorless methods, right? Everyone got the vectorless methods. Do you need to know the vectors which are involved? You study the vectorless methods. Do you need to know the vectors which are involved? If yes, let me know. Now, right now. If at all you need to know right now. Elution is a method in which you take the DNA out of the gel electrophoresis thing. Yes, ma'am. Okay, perfect. So listen to me carefully, okay? Okay, the vectors that you use, there are two types, okay, there are two types. First thing, the basic vector that we have is a plasmid. Okay, plasmid is something that we use every single now and then, but apart from that, you also have the plasmid of Agrobacterium tumefaciens. Okay, there is a organism that is known as Agrobacterium tumefaciens. Now, why do we need it? So, just like how you had biolistics for plants, similarly, in vector method, you have Agrobacterium tumefaciens for transfer of things to the plants. Why is it acting like a vector, especially for the plants? Because of one very important reason. Suppose this is the bacteria that is out there. Okay, this is the bacteria that we have, and over here, it has its own plasmid. You know the name of this particular plasmid? It is known as TI plasmid. Why TI plasmid? Because it induces tumor. So I'll say that it is what? Tumor inducing plasmid. What is it? It is going to be tumor inducing plasmid. How can it induce tumor? So basically the thing is that it is also having a sequence right over there. You call this as tDNA. tDNA is also known as transfer DNA. It is also known as transfer DNA. This particular transfer DNA is something that causes the tumor. How? So basically when the bacteria goes and attacks a plant, okay, goes and attacks a plant right up over here. So what exactly happens is that it is going to transfer the plasmid, and in the plasmid what do you have? You have the tDNA. This causes what? This causes tumor in the case of plants. Now we guys are very much brainy, right? We think that we are brainy, but yeah, we are a little bit brainy, okay? So what happens is that we have taken the machinery. We thought that if it can do this, why not remove the tumor affecting part over here? Why not remove the tDNA part over here and insert the gene of interest? So we insert what? We interest our gene of interest, and we let this organism infect the plant. But now when it is infecting the plant, what is going to happen? Basically it is transferring our gene of interest. Now this gene of interest can be pest resistant gene and things like that. So now the plant is literally very, very happy. Is this thing clear to everyone? Is this thing clear to everyone? Everyone who is watching this video, very, very important. Yes? Done? Ma'am, this video contains how many marks? Dear, I'm trying to complete 4 chapters over here, so I have to calculate. Yes, anyways. So the previous video, this video together will give you 70 plus 70 is what I'm assuming. Okay, anyways. Yes, perfect. Now we have one more thing, okay? Now we have one more thing. Apart from plants, we have animals also. We have animals also. If not 70 plus at least 60 marks, definitely you should be getting, okay, because MCQs we still have to do. Okay, now what do we have? Oh, okay, animals. In the case of animals, you know HIV is a retrovirus. How does it work? It has its RNA. It basically infects us and it transforms into a DNA, and that is, it uses our machinery to do the working. Similarly, just understand, basically, isn't it transferring something into our body? Yes, it's genetic material. Similarly, if at all in this particular retrovirus, we remove the things that is harmful for us. Okay, we remove the things that is harmful for us, and basically, if at all we put our gene of interest over here, gene of interest over here, what is going to happen? It is still going to infect what? Still going to infect the human being out there, and this time it is passing our gene of interest. So isn't this useful? Obviously yes. And that is why we say for animals, what do you have? You have retroviruses, and you always mention this as disarmed retroviruses. What do you mention this as? Disarmed retroviruses. Okay, I really want to compliment you. Real compliment, no problem. Just that you need to study also. Okay, guys, is this clear to you? Everyone? Hi, Pranav. If yes, give me a thumbs up. Cooked in biology. Anand, come on. Sanjeevani, thank you, guys. Okay, so we go ahead with the next thing that is there. That is the next question. I hope now the vector and the vectorless method is clear to you. Over here, I have the first question, that is, origin of replication in vector is an essential site required for gene cloning. In a vector, whether it be a plasmid or anything, my dear children, you need an origin of replication site. Why do you need an origin of replication site? Why do you need an origin of replication site? Everyone think about it. By then, I am going to do something very important. Okay, my energy pulls too. Thank you, that's so sweet of you. By the way, guys, just give me 1 minute, okay? You try to find the answer for this. Give me 1 minute. I'll be right back. Okay, perfect. Replication starts. This is why you want this to be done for gene cloning. Okay, so many people have different answers. Listen to me carefully. Origin of replication is a specific sequence from where replication starts. You know, DNA polymerase has to come and attach over here. Suppose you have your gene of interest. If at all there is no one to attach over here and do the replication and make a copy of your gene of interest, is it of any use? No. That is why we need this. Origin of replication is a specific sequence from where replication is going to start, allowing the vector to replicate inside the host cell and produce what? Multiple copies of the inserted DNA, or basically your gene of interest. So controls the copy number, initiates the replication. Ram Patel. Yes, exactly. Okay, and yes, you are absolutely correct. It also controls the copy number, as in how many copies have to be made. Okay, that is super correct. Now the next question that we have is, selectable marker in a vector is essential. Why do you think selectable markers are super important in a vector? What do you mean by selectable markers? For example, you have ampicillin resistant gene, you have tetracycline resistant gene. Why do you think they are important? Why do you think they are actually important? Any idea? It allows the vector to produce multiple copies of itself along with the inserted foreign DNA. Exactly. The origin of replication is a vector because it's specific DNA. Exactly. You're absolutely correct. Lich kills and Gia. Everyone who gave me the answer, you're perfectly giving me that. Okay. Now I'm asking you the next question out there about the selectable markers. Why it is essential for you? Elimination of transformants. Elimination of transformants? No, elimination of non-transformants, right? Right? Eliminates the non-transformants. Listen to me carefully. This is important. Okay, this is important. Probably this might give you an idea about a lot of things while I explain you this. So I have a scenario over here. Suppose there is one particular bacteria. This is another bacteria. Okay. What exactly happens is that usually this organism does not. Usually these organisms that you see does not have any resistance to any of the antibiotics out there. It does not have it at all. Okay. Now what we are doing, we are going to use the plasmid. This particular plasmid is PBr322. This particular plasmid is PBr322. And suppose I have, so first scenario is like this. First scenario is like this. Listen, if you understand this, you understood half of the biotechnology. 3 bacteria I have, 3 of them are same bacteria, they do not have any antibiotic resistant gene. So what happens when I basically insert PBr322, when the vector is getting inside, okay, I will say that it has transformed, it is something new now. So over here, the vector got inside, now you have PBr322. This is also PBr322, but what is there? Here I have put my gene of interest. This PBr322 has my gene of interest. So I will call this as what? Recombinant DNA. This is what? Recombinant DNA. Why? Because it is not normal PBr322, it has my gene of interest. That is why recombinant, new combination is happening over here. This is a normal PBr322. So in one particular in one particular bacteria out there, it does not take the plasmid. In another bacteria, it has taken in the plasmid, okay, and in this particular bacteria, it also has taken in the plasmid, and this particular plasmid has my gene of interest as well. What do you call this cell who has not taken up anything? What do you call this cell which has not taken up any plasmid? This is a non-transformant. Transformant. Transformant means, for example, from today, I am a new person altogether. There's a transformation happening out there. There might be some changes. But here, Baba, there is no change. The bacteria is just like that. How did we get to understand that? Because I was not able to grow this bacteria in an ampicillin medium. Why ampicillin? Why I am not able to grow? Array, it does not have the ampicillin resistant gene, so it will die off. It will die off. If at all it had the plasmid, probably it would have stayed alive. Why? Because this particular plasmid has ampicillin resistant gene and also have tetracycline resistant gene. What about this plasmid? This also have ampicillin resistant gene, but it does not have tetracycline resistant gene. This is recombinant plasmid. That's why. So over here, this guy is not able to sustain at all. Why? Because it's a non-transformant. And how are we able to identify that? With the help of the selectable markers, that is ampicillin resistant gene and tetracycline resistant gene out there. So this is not transformant, and this is going to be a transformant, and this is also going to be what? A transformant. Both of them have got the new plasmid out there. Now among them, which one is a recombinant? If at all I call this A, I call this B, and I call this C, you have to tell me which one is a recombinant. Which one is a recombinant? Any idea, guys? Different type of questions gonna come for tomorrow's examination. Ma'am, you are Hindu or Muslim? I am basically a human being. Okay. Yes. Okay. C. Everyone is saying C. Exactly. C is the one. C is the one. Right? C is the one. Why C, Baba? Over here, C has the gene of interest. This is going to be a transformant because it has taken up a new plasmid. It is also going to be a recombinant. Why? Because it has my recombinant gene as well. This is super important. Got it? So I hope everyone have got it. Muhammad Kaif Fatan is saying foreign gene is there. Exactly. So guys, is it clear for everyone, or do you need a repetition? I'll tell once more because I know it can be. Yes, I can surely repeat it. Okay, listen to me carefully. Okay, there are 3 bacteria that is out there. Okay, and I have basically put the plasmid DNA around here. Some plasmid DNA took the gene of interest and all. You call this as recombinant plasmid altogether. This plasmid is a plain plasmid. It has not taken up anything. This plasmid has ampicillin and tetracycline resistant gene. Here it will only have ampicillin. In the tetracycline site, I have inserted something else, so it is not resistant to tetracycline anymore. Anyways, when I put them together, what happened? this cell did not take the plasmid, any of the plasmid, it did not take anything. This cell took the plasmid. Which one? This plasmid. Okay. This cell took the plasmid, that is what? The recombinant plasmid out there. Okay, fine. How do I understand which one has taken what? Basically, the first thing that I am going to do is, I am going to treat it with a medium of ampicillin. I am going to put all of them, my dear children, in a medium that has what? Ampicillin. You know, ampicillin is an antibiotic. The one which dies is the one that does not have the plasmid DNA, because if at all you have the plasmid DNA, you have the ampicillin resistant, it will not be dying, right? So I got an understanding that when I grew them in ampicillin medium, this guy A, it died. It died. That means what? It does not have the plasmid. It does not have the plasmid, does not got the plasmid. So do you think it has taken up any change out there? Do you think it has got transformed? No, Baba, it has not got transformed. So I'll say it is what? A non-transformant. Whereas when you talk about these two guys, here they are able to survive. Why? Because they have the ampicillin resistant gene. How did they get it? Because they took the plasmid out there, right? So I'll say these two are what? Transformants. They got transformed. At least they got it. Now, the next thing that I have to do is, basically, I have to find out among them who are the recombinants. I have to find out who are the recombinants out there. Transformants, so I got to know. These two are the transformants. How do I find the recombinant? It's very simple. You need one more plating. So, whenever you take PBr 322, you have to do two plating. One in ampicillin and one in the next thing, that is tetracycline. So, my dear children, I am going to remove this guy, because anyway this guy is non-transformant. Until here, is it clear to everyone? Until here, is it clear to everyone out there? Children, is this clear to everyone? Yes, thank you. Now listen to me carefully. Over here, I am going to remove this guy because this is a non-transformant. Let me remove this. Now what we are left with? We are left with two transformants out there, and basically I have to find among them which one is the recombinant, which one has my gene of interest. That is the next question. So what do? I am now going to grow I am now going to grow them in a different media. What is this media? This is going to be tetracycline media. So this is my plasmid, sorry, this was my one cell, this is another cell, this was the B cell, this is the C cell. Over here, there is the plasmid PBr 322 without the recombinant DNA. So I'll say it is ampicillin resistant. It is also tetracycline resistant. Here I have my PBr 322, but this is a recombinant PBr 322. It will be only having ampicillin resistance. What about tetracycline resistant? It is not tetracycline resistant because it have my gene of interest. I know that because it is being drawn over here. But how do we identify it? We put it in tetracycline medium. You tell me, in them, which one will be able to divide and grow? B or C? If at all I am growing it in tetracycline medium, which one will be able to grow? B or C? B. Okay. Jetharam Patel says it is B. Mansi says it is C. Will C be able to grow if at all I am putting in a medium that has tetracycline? B. Yasin says B, and Riddhi also says, everyone. G also, Ayu also, Karthik also. Everyone says B. Exactly. Why B? Listen to me. It is resistant to tetracycline because it has the gene, it has the superpower. But here we have put the gene of interest. It is not resistant. So what do we understand from this? That here it has not taken up the gene of interest. This is going to be what? A non-recombinant. It is the one, that is non-recombinant. It does not have my gene of interest. Whereas the one which is not able to grow and divide, it is the one which is going to be the recombinant. So we are going to exclude the ones which will be able to grow. We are going to take the ones which are not able to grow, and then we are going to culture them. So this is ulta. So we usually think the ones which are able to grow, will take them. No. So ones which are not able to grow, we are going to take them over here. That's the logic. Is this particular thing clear? So whenever PBr 322 is there, you need two plating. Transformants and recombinants are different. Transformants means taking up any new DNA or anything from outside. Transformants are the cells which are going to take up the new DNA from outside, or basically any extra foreign particle, foreign DNA from outside. Recombinants are the ones which takes up what? The recombinant DNA, not just any foreign DNA, but the recombinant DNA out there. Got it? If it's clear, just give me a quick thumbs up, or you can give me heart also. Either way, whatever prefers to you, please give me that. Gaming, is this enough to get 60 plus? Yes, that is what I believe, dear. Gaming legends. Yesterday's class you were there, right? That plus this, I'm covering the other chapters also. Okay, perfect. Thank you, guys. So why I explain this in more detail is that you will see a separate question. There I will need to explain less. Okay, anyways. Yeah, perfect. Now, selectable markers, antibiotic resistant gene, etc. help identify and select the transformants from the non-transformants, thereby enabling detection of recombinant DNA. This is important question also. With the selectable markers, do you get recombinants or transformants? The first primary thing is that you will be able to find the transformants. Then the next thing is recombinants, right? First thing is transformants, not recombinants. Do not go and write that. I have seen an MCQ question where it has been written that with the help of selectable markers, will you be able to find the transformants or recombinants? It is basically first thing, transformants. Then comes recombinants. So if at all that question comes, please go ahead with transformants. Do not go ahead with recombinants. Okay? Is this clear? Important. Now the next question, vectors should have very few preferably single recognition sites. Vectors should have few or single recognition sites. What is that, dear? Okay. What do you mean by that? Why they should be having only single recognition sites, vectors? Any idea? Hello, gamer girl. Most fixed questions highlight my dear. I have given you a separate question thing for that itself. Yes, and here, yeah, we are dealing with that. Replication takes place at that particular site. Exactly. No idea, ma'am. No idea? Okay. Vectors should have very few, preferably single recognition sites. That means that basically, I just told you about PBR 322. We studied about PBR 322, right? What did we study about PBR 322? The question is, why does BAM H1 does not have multiple sites over here? Why does this particular thing have only one site? Because suppose this particular guy has a site over here also. What is going to happen? BAM H1 will make a cut over here. It will make a cut over here. Array, the whole plasmid is now cleaved, right? It will be cleaved. So every single restriction enzyme which is there, it will have only one cloning site. One cloning site per restriction enzyme. You can have multiple restriction enzymes, but per restriction enzymes you should be having only one. You get it? This is super important. Now over here, a vector should contain only one recognition site for a particular enzyme, so that it cuts the vector at only a single location, allowing proper insertion of foreign DNA without fragmenting the vector. You do not want to kill the whole vector out there. Did you get it? This is a twisted question in a different format. I hope this is clear. Yes? Yes? The largest genus of dystrophin. Okay. Perfect. Thank you, guys. Thank you, Ayu and Chandrasekhar. Okay. Now, selection of recombinants by antibiotic inactivation is cumbersome. Selection of recombinants. Selection of recombinants by antibiotic inactivation is cumbersome. Cumbersome means what? It is difficult. Why do you think it is difficult? Because we just saw that there are two platings required for finding the recombinants, right? In first plating, basically you found the transformants. In the second plating, basically you found the recombinants. So obviously it is very difficult with the help of the antibiotic resistant gene and stuff like that. There is another method, that is lag-Z gene. So there is another plasmid altogether, which makes things very easy. Right, there is a lag-Z gene and stuff like that. PBR 322, which has antibiotic resistant gene as selectable markers, it becomes difficult because you need two plating. Double plating is needed over here. Okay, so what is happening? This method requires plating transformants on two different antibiotic media to identify recombinants and non-recombinants, making the process time-consuming and laborious. As in, first plating you do to find the transformants, the second plating you do to find the recombinants. That is why it is considered to be cumbersome. Got it? Is this clear to everyone? Hello Marlika, everyone guys. What are you doing over here? Chandrasekhar ma'am, I'm a big fan of you. Oh, thank you. Even I'm a big fan of you guys also. Yashwant, hello. Thank you. Okay, Ritika and Ayu and Riddhi, perfect. Now next thing, chilled ethanol is used in DNA isolation. Baba, DNA isolation, if at all you want to isolate the DNA, you use chilled ethanol. Why do you use chilled ethanol? Why not anything else? Why not anything else? Any idea? Come on. Ma'am, had your lunch? Yes, dear, today I had my lunch. What about you guys? I hope you had. To precipitate out the DNA, why not you? Exactly. When you add chilled ethanol, what is going to happen? This is insoluble. The DNA is insoluble in chilled ethanol, which is why when something is not soluble, what is going to happen? It will precipitate out. It will come out like that. That is the thing. Okay, so please remember, this is very, very important. DNA is insoluble in chilled ethanol, so it precipitates out. This term is important. Okay. It's insoluble, so it precipitates out the solution, making it easy to separate and spool out the DNA. These are the questions which are there. This is from your question bank. I took this question. Why? Because I think there are chances for these type of questions to come in. Okay, perfect. Clear? Is this question or all these questions clear to you? If yes, give me a thumbs up. We'll move on to the next question. Ma'am, are model and preparatory enough, ma'am? No, dear. Please go ahead with other things also, not just them. Yes. I'm reminding you to hydrate your body. Thank you, Varun. I'll definitely do so. Okay. Now, the 51st question is there. Given below are the diagrams of plasmid A and B. Observe meticulously and answer the questions that follows. Okay, this is plasmid A. What is plasmid A? This is PBR 322. Okay, what is this plasmid? This is another plasmid, plasmid number B, which has LAG-Z gene. Do you know about these plasmids? Okay, can you repeat E? Sorry, dear. Okay, just a second. Repeat E. Chilled ethanol is being put, okay, when you put chilled ethanol, what happens is that DNA is insoluble over here. So, which is why it will precipitate out. We can get it separately and we can take it out. Okay? Yes. Fine. So, you guys know about it, right? Now, listen to me. Which plasmid is or are you going to select for cloning and why? So, among these two plasmids which are right in front of you, which ones are the ones that you are going to take for cloning purpose? Any idea? Any idea? Among them, this is A, this is B. A, Hassan says A, Varsha says B, Simran says LAG-Z1, this one, right? Okay, Sanjana says A. Saida will, okay, so I'll talk about that, just a second, okay? Warren, everyone mostly are saying A. Why A? Why A? Why do you think A? If you are saying A, that means you do not know how it works. You do not know how it works. That is why you are saying A. Listen to me very carefully, okay? This is LAG-Z wala plasmid out there, okay? It has a whole different, fan following for it. I'll tell you why. So, suppose this is a plasmid out there that has LAG-Z gene. The LAG-Z gene, in lac operon, what does the Z gene do? It codes for beta-galactosidase, right? Galactosidase. Now, over here, in this particular medium where I have the LAG-Z gene, my dear children, or basically I have the plasmid, if at all I put a coloring substance, I put a coloring substrate over here. For example, there is a thing that is known as X-gal. If I put X-gal, what is going to happen? Because of this particular enzyme acting on it, you will get a blue color thingy. You will get what? Blue color thingy out there. Fine, fine. Now, if on the other hand, I am putting my gene of interest over here. If I put my gene of interest over here, what is going to happen? Will it produce beta-galactosidase? No. So even if I put X-gal, which is the coloring substance, do you think I'll get a color change out there? Do you think that is going to act on anything? No, because I do not have this guy. What will act? So which is why I get what? White color thing out there. I will get white color thing. There is no blue coloration. There is only white coloration. Got it? Now listen to me carefully. Over here, in this particular case, how will I find my recombinants? How will I find my recombinants? The ones which has, suppose this is a cell and this is a cell. This particular cell has a normal plasmid out there with LAG-Z gene. It has the LAG-Z gene. This one has the plasmid but with my gene of interest. With my gene of interest. What do I need to do? In this particular area, I just need to add the X-gal. the coloring substrate. When I add the X gal, what is going to happen? Over here at this particular corner, it is going to release beta-galactosidase and will be changing a color. Will be changing the color as in this tide will be turning blue in color. It will be turning blue in color. Now what about that side? Over there, there is no enzyme. There is no enzyme being produced because it has my gene of interest. Which is why what is going to happen? They will not have a color change. Here you will get white color. So within the same plating itself, I do not need two plating to do. One plating enough is enough. Why? Because is enough. Why? Because over here in one plating itself, the ones that has the gene will be giving me blue color. The one does not have that gene will be giving the white color. I'll be taking the white ones for doing the next process out there. Or basically, I'll be taking the white ones to and I'll put in the bioreactors so that they can grow. That's all. Only one plating is required over here. You get it? So the answer for this question is going to be plasmid number B. Okay. Ma'am, please tell your team to. It's important questions. Fixed questions are done by Ma'am Da. She put it yesterday only, I think. Okay. B. Okay. Now, hello, Haripriya. Perfect. And this is known as blue-white colonies because all the cells which will have the color blue, blue colony will be formed, and there it will be forming white colonies out there. Okay, perfect. Next thing, what is insertional inactivation? What do you mean by insertional inactivation? What is the meaning of insertional inactivation? Insertional inactivation. Insertional inactivation means a gene which is there, it is going to stop working. Why? Because we are inserting a foreign DNA out there. We are inactivating the older gene. This is known as insertional inactivation. So how do you write it? It means a gene stops working because a foreign DNA is inserted into it. Example, in plasmid B, LAG-Z gene stop producing the enzyme beta-galactosidase after insertion of foreign DNA. So when you give an example and you explain, it becomes all the more good. Okay, it is very, very attractive way of answering things. Okay, perfect, done. Yeah, yes. Now, since so many guys of you are asking me, ma'am, tomorrow morning there is a class or not? So I was supposed to record a video of last minute revision. That recording did not take place because I did not get any time to prepare a PPT. Okay, so what is going to happen? I am going to come live. Okay, I will be coming live, but not at 4 o'clock in the morning. It will be quite unsafe for me. So I'll be coming at around 5:30 if all goes well. Yeah, 5:30 in the morning I'll be coming for last minute revision live, where I'll be like going like 10x speed for the MQPs. The MQPs, you know, that is important. Everyone says that, and everyone is super scared and anxious about MQPs. So tomorrow morning we will revise with the help of MQPs. I'll go on really fast such that you can cover everything up. Does that work? So your MQP tension will be sorted out there. So it's for those of you who have covered MQPs at least once, because as I said, the speed will be really fast. Okay, for KCET, I'll be taking the classes after this. After boards, we will be starting off with KCET. Okay, dear Chinmay? Yes, perfect. And tell your friends also to be there for the live. It will be quite refreshing for all of you guys. Yes. Ma'am, explain the blue again. The blue again. So the blue one means that it has the LAG-Z gene. LAG-Z gene will be there. Beta-galactosidase is going to produce. This enzyme acts on the coloring substrate, gives a blue color thing. So over here, if it is giving the blue color, that means it is still having the LAG-Z gene. It is not replaced by a gene of interest. Because if it was replaced by gene of interest, it would have given us the white colonies out there. Okay? Ma'am, when does it start? 5:30 a.m. in the morning, it will be starting. Okay? Now, will the number of culture plating be the same or the different? Now you tell me, you know the answer for this. Will the culture plating be same or different? Comment. No, do not just read only the model question paper, okay? Thank you, people entertainer. You're most welcome. Okay, same, different. Okay, Riddhi says different. Sanjana says same. Same, macha. Sunita says different. Okay, Nicola says no, different. Varsha also says different. Guys, it is going to be different. Okay, it is not going to be the same, right? In plasmid number B, plasmid number A, which is PBR 322, you need two culture platings, right? One plating in ampicillin for finding the transformants, another plating in tetracycline for finding the recombinants. So over here for plasmid A, ampicillin a tetracycline system needs 2 plates, one with ampicillin for finding the transformants and one with tetracycline for finding the recombinants. What about plasmid B, lag Z system, needs only 1 plate. Okay, needs only 1 plate with X-gal. White one give recombinants, blue ones means non-recombinants. There is no of transformants and stuff like that is not been included over here. So this is equal. Done. If this particular question is clear, okay. If this is clear, just give me a quick thumbs up. Perfect. And I hope everyone is understanding what I'm trying to teach, right? Do not forget to hit the subscribe button. Whoever is watching this video, who is new to this channel, hit the subscribe button right now. Okay. Now what do we have? We have the next question, that is elaborate PCR. PCR means what? Everyone, come on. PCR means what? PCR stands for what? Polymerase chain reaction. Exactly, the spelling is wrong, okay. Polymerase chain reaction, right? And why do we need it? For amplification of DNA, for increasing the number or basically the count of the DNA out there, you need polymerase chain reaction. And it works very simple. Listen to me very carefully. You have the DNA, right? You have the DNA strand. You want to amplify it. The first thing that you're going to do is denaturation. Okay, the first thing that you're going to do is denaturation. By the way, before I get into that, I want to tell very important things. What are the tools that you require over here? If you want to make more DNA, obviously you are going to need what? The DNA polymerase. You will need DNA polymerase. Now this DNA polymerase should be very different from the other usual DNA polymerase. Why, Baba? Because this whole process of PCR happens at very high temperature. It can go up to 95 degrees Celsius also. So we actually need a thermostable DNA polymerase. What do we need? We need thermostable DNA polymerase. So where can we get that from? We get that from an organism that is known as Thermus aquaticus. Thermus aquaticus, that is where we get the DNA polymerase. So this organism lives at very high temperature. What do we do? From this organism we take the DNA polymerase, and which is why we call this unique DNA polymerase as Taq polymerase. Just a second. You call this as Taq polymerase. So the first thing is done, we need the DNA polymerase, and the name of that is known as Taq polymerase. The second thing that you require over here is what? You know, the DNA polymerase cannot start off the polymerization alone. It always needs the primer. It always needs the primer, right? It needs a primer. What do you call this? Is it RNA primer or DNA primer? It is made up of RNA or DNA. The small segments of things that you need before, such that the DNA polymerase can extend it. I'm asking you, is it RNA primer or DNA primer? Guys, come on. RNA, RNA, RNA. Perfect. That is how we are going to lose our marks. Okay, it is DNA. Listen, normally within our body, when replication happens, that's when we have RNA primers. That's when we have RNA primers. In the case of PCR, there is no RNA primers. In normal replication, RNA primers. But over here, DNA primers are going to be utilized. Remember that. So what do we have? Deoxynucleotide primers are there. DNA primers are going to be used. Okay, apart from the primers, what are the things that we need? Apart from the primers, it's okay, do not retract your message. Guys, do you think I am someone who is going to judge you guys? No. Tell me, this very important thing. Apart from DNA primers, apart from DNA polymerase, what else do you need? Taq polymerase, I told. Taq polymerase is done. Next thing that you might need. Any idea? Amplified fragment. We need the unit such that the DNA polymerase can attach, no? We need the nucleotides. What do you call these nucleotides as? What do you call these things which comes together to form the DNA? Deoxyribonucleotide triphosphates. What do you call them? Deoxyribonucleotide triphosphates. DNTPs is what you call them. Okay, you call them as DNTPs, deoxynucleotide triphosphates. So in that triphosphate, 2 of the phosphate goes, 1 phosphate remains, and that is how you form the bonds. This is super important. These are the things that you require. Please do not forget them. Super important. Now we go ahead with the process, but before I go ahead with that, please take a screenshot of this particular thing. Otherwise, sometimes my hand touches, it goes off. Okay, yes. Ma'am, when will the lecture end? This particular lecture is going to end by 6:30. Okay, okay, let's go. Now, first step is denaturation. What do you mean by denaturation? You have the 2 strands out there. These 2 strands, one which is in 3' to 5' direction, and other which is in 5' to 3' direction, are going to separate. They are going to separate. This is known as denaturation. It happens at around 90 to 95 degrees Celsius. Very high temperature. Done. So we are breaking the bonds up over here. Okay, because we want to make a new strand. The second step that is there is known as annealing. And this is where most of you get your answers wrong. What is annealing? Annealing means basically we are going to add the primers. Over here you need to pay attention. This is 3' end, this is 5' end. Okay, and this is again 5' end, and this is 3' end. Right? Where do you think the primers are going to attach? This is the process of attachment of primers, but where do you think the DNA primers are going to attach? Which end? Which end? Tell me. The DNA primers attaches as which end? 3' end or 5' end? Nana. 3' end. Sudhita says 3' end. Everyone says 3, 5. Someone said 5. It is at 3' end, Baba. Here the DNA fragments are going to come and attach. So your primer which is there, it attaches only at the 3' end. Now what do they do to confuse you guys? They make the picture ulta. In your NCRT, 5' is over here, 3' over there. Okay, so people think, oh my god, ma'am, what are you doing? It's the primer should be there. No, look at the position of 3'. Wherever 3' is there, opposite side you will get the primer. Okay, you will be getting the primer. This is super important. Why do you think the primer is attaching over there? Because in the next step that is known as extension, the next step which is known as extension, what is going to happen? Your tag polymerase is going to extend the DNA fragments. In which direction? All the DNA polymerases extends in 5' to 3' direction. So over here, this is going to be the 5' end, and it can just extend like this. That is why it attaches at the 3' area. You get it? DNA polymerase, RNA polymerase, everyone does like that only, Baba. Okay? Okay, did you get it? I hope this helps. Yeah, so do not make this mistake. Look at the 3' spot and then only go ahead. And please remember, this happens almost around 50 to 65 degrees Celsius. Over here, extension happens almost around 75 degrees Celsius. All of it is happening at high temperature, and this is for amplification of DNA. One more very important thing. If a question comes, this is related to biotechnology and its application, but still I am asking about that. Explain once more. I can do that. Yes. Ma'am, are you coming to Deeksha tomorrow? Which Deeksha? So we are at Deeksha VLC campus. Okay, that is DVLC campus. So that is why we get to see children who come, Vedanta, children who are from Vedanta college. I've seen their tags over here. So they usually come, we meet them. Yeah. So I will be there in the DVLC campus. Definitely, for sure. Don't kill me if at all there is someone who's, ma'am, I could not do the MCQ because of you. Don't do that, okay? I will be scared, I will not come. Okay. So, yes, explain once again. I'll do that. Listen to me very carefully. That is, first step is denaturation. Denaturation means basically you're going to separate the DNA fragments. Why? Because we want to make the copy. It happens around 90 to 95 degrees Celsius. Second thing is annealing. Annealing means you have to add the primers because the DNA polymerase needs the primers to extend. Now, where do you add the primers? At the 3' end. Here at the 3' end, here at the 3' end. Why? Because the new strand that is going to be formed will be formed from the 5' to 3' direction. 5' to 3' direction. That is why we are adding in the 3' end. Right? Now the next step is extension, where it finally extends throughout. And that is how it goes. That's all. Simple. Okay? Now listen to me very, very carefully about this particular thing. Question that is there from biotechnology and its application, that is, it goes like this. If at all there is a pathogen, if at all there is only very less amount of pathogen, which process do you use to identify? Okay, which process is something that will help you identify the pathogen out there? This is a very important question. People entertainer, dear, can you, everyone, is it okay? Is it audible? Can you increase the volume by any chance, if at all it works? Guys. So, yes, okay. So if at all you have very less amount of pathogen, guys, what do you use? Very less amount of pathogen is there. What do you use? Come on, tell me. Which process do you use? I'm asking you a question. And no one is giving me the answer. Why is that? Didn't you understand the question? Children, if there is for example, in my body, there is only little amount of pathogen which is there. But I still want to find who is this particular pathogen. With little amount also, how will I? PCR, exactly Sanjana, exactly Swati. It is what? PCR. PCR method is utilized for finding this particular thing. Very, very important. In your NCERT, biotechnology and its application chapter, it is given. Do not miss it out. Okay? Now, Anik, can you repeat, ma'am, the question you're asking, dear? Ma'am, first tell the fixed question is already given to you, dear. I'm solving few of them also here. Yeah, and I regret choosing bio, ma'am, because I've chosen bio, ma'am. I could have chosen CS. Do not worry. Listen, go join the WhatsApp channel. Listen to me very carefully. Everyone who is worried about bio, press on this particular thing. You see the first link that is out there. Okay, whether you have to join the WhatsApp channel, click on that particular link. Okay, you do not have to put any number and all. Just click on that particular link. I have given you a message in there. I typed it. I have typed this whole message out there. Make sure you read it. You will get an understanding of what video is best for you in this particular scenario. Okay? Yes, perfect. Last exam, yes. Okay, perfect, guys. So PCR is a method that you use for finding small amount of pathogens. And how do you write this answer? PCR is a technique used to amplify a specific DNA segments in vitro in the laboratory condition using two primers, DNA polymerase and the nucleotides. What nucleotides? DNTPs. Okay. Now, denaturation, DNA is heated at this temperature to separate the two strands of double-stranded DNA. Annealing, temperature is lowered so that the primers bind to the complementary sequence on the DNA. Extension, Taq polymerase from Thermus aquaticus extends the primers. These steps are repeated for 30 cycles, producing 1 billion copies of the DNA segment, which can then be used for cloning for further things. Okay? Adi Vidya Math. Hello, JJ ma'am. Hello, dear. And Sham, ma'am, you are beautiful brain. Oh my God, thank you. Okay. So guys, come back. I hope this is clear. We go ahead with the next thing. So children, with this we are going ahead with the next chapter. But before that, I also want you to have an idea about two things. Everyone who is here, tell me who all wants to have a brief idea about gel electrophoresis and also isolation of DNA. Gel electrophoresis and isolation of DNA. If at all you do not have an idea of it, and if you want me to tell, please let me know right now. This is important from this particular chapter. Me, Kid Zone, okay, Riddhi, okay. Everyone says me. Okay, so listen to me, gel electrophoresis, about gel electrophoresis. Okay, first thing. What do we do over here? Okay, no, wait. I will start with the isolation of DNA first. That is a step-by-step process, no? So, isolation of DNA. How do we isolate the DNA? Suppose it's a fungal cell. In the fungi, first of all, here inside the nucleus you have the DNA, right? But the problem is that the fungi has a cell wall. Fungi will be having the cell wall, and the cell wall is made up of chitin. So what do you need to do first? First you need to break open the cell wall and need to get entry inside. So for that, what do we need? We need the enzyme chitinase. So chitinase is an enzyme that will break open the cell wall, and that's how we get into it. Fine, this is the first thing. Second thing, suppose it is a plant. In plant, Baba, again the cell wall is there. Agreed? The cell wall is there. What is the cell wall made up of? It is made up of, guys, what is the plant cell wall made up of? Any idea? Any idea? It is made up of what? Cellulose. Okay, and if at all we want to break it open, what do we need? We need cellulase. We need cellulase enzyme. So again, this is a thing that we need. Yeah, perfect, guys. I see your answers. Awesome. Now we have bacteria. We have bacteria. In the case of bacteria, again, they also have cell wall. Their cell wall is made up of peptidoglycan. What is it made up of? peptidoglycan. Now what do we need for the breakdown of peptidoglycan? You guys cry when something gets inside your eyes, right? Why? Because, my dear children, our eyes naturally produces lysozyme. Lysozyme, they are there in the tears. Okay, these lysozyme are nothing but enzymes that breaks open the cell wall of the bacteria, kills the bacteria in. So lysozyme, cellulase, chitinase, you should know for each of them. Chitinase for fungi, cellulase for plant, and lysozyme for bacteria. Done? Okay, now we have got an entry into the cell. What after that? When you get entry into the cell, listen, your DNA is not alone out there. There is also their friends RNA and protein. But you just want the DNA. So what do? You have to degrade the RNA out there. What do we do for degrading the RNA? We treat it with RNAs. We treat it with RNAs. For example, we want to degrade the protein out there. What do we have? We have proteinase. We have proteinase. So all this small information is super important. And finally, you have the DNA. Will you treat the DNA with DNase? No, Baba, you need the DNA. You cannot do that. Now, finally, these machals are also there. The broken fragments of RNA, the protein, everything is there. You want to separate it from the whole DNA. What do? You put chilled ethanol. When you put chilled ethanol, what is going to happen? The DNA precipitates out. Why? Because it is insoluble. It is insoluble, and that is how you do the isolation of the DNA. Is this particular thing clear to everyone out there? Is this particular thing clear to everyone out there? And this process is exactly known as what? Spooling. You take it out. Okay? Philip Johns, hello Jasmine ma'am, do you remember me? Should I lie? I should not be lying, right? So I'm so sorry if at all I'm not able to remember all of a sudden, probably with some instance, if at all, probably should be. I'm so sorry if at all you were my student or something like that. Okay? Yes. Listen. Yeah? It's okay. I'll tell you something honestly. That is, I am a person who forgets words. I'm starting to forget words. I'm literally scared I must start to forget human beings altogether. So please forgive me if at all I had. But I try my best to remember. So yes. And okay. So this is particularly clear. So we move on to the next thing, and that is what? And that is what? You have the next thing, that is gel electrophoresis. So before that, you use restriction. Oh, okay, I'll tell about that also. Listen. After you have isolated the DNA, you use restriction enzymes to cut the DNA. You use restriction enzymes to cut the DNA, right? So how do we do that? Basically, you have two restriction enzymes. One is endonuclease, one is exonuclease. Endonuclease are the ones which goes deep into the DNA, makes a cut in the phosphodiester bond. What? Phosphodiester bond. That is how things works. Okay, endonucleases. Exonucleases are the one that makes the cuts towards the end. And the most famous endonuclease which is there, it is known as EcoR1. It is known as EcoR1, where here E stands for Aceracea, the genus name. CO stands for this first two letters of the species name, that is coli. R stands for the first letter of the name of the strain, and 1 stands for the number of discovery. Okay, and the site for EcoR1 is known as 5-GAATTC. Okay, 5-GAATTC. GATTC. This is super important. Okay. Remember that. And apart from it, also remember the first restriction endonuclease to be discovered was HIND2. It was HIND2. In PBR 322, you have HIND3. Here it is HIND2, which is the first discovered endonuclease out there. It also has a restriction site of 6 base pairs, but the restriction site is not been mentioned. Okay, it is not been mentioned. I hope that is being clear. That is clear to you. And yes, with the help of endonuclease, you get sticky ends and things like that. And with the help of endonuclease, you get fragments. Yeah, all of these things. So please know the the basic things about exonuclease and endonuclease, the differences which is out there. I have told you the basic thing. And so sometimes children get confused. Ma'am, in your book it is written that EcoR1 is the first one where restriction enzymes were isolated. Listen, EcoR1 was the first one where they've got an understanding that restriction enzyme is there, that some enzyme is there that adds methyl group and everything. Okay, but they did not discover the whole enzyme as in totally. So it was hinted to where it was finally been discovered. I hope this is clear. Okay, guys, what all questions do you have, children? Tomorrow you have examination, and okay. Okay, done? Yes, perfect. Now what do we have over here? Yes, so restriction enzyme has also been done, and please know about the sticky end formation and stuff like that. What do we have next is gel electrophoresis. Yeah, so in gel electrophoresis, what do you have? You have wells, my dear children. You have small wells right over here. What do you put over here? You put the DNA. You put the DNA that you have just caught. Okay, you put the DNA that you have just caught right over here. Now what happens when you put the DNA? Basically, here you have cathode, and cathode is negatively charged. Here you have anode, and anode is what? Positively charged. Over here, from the cathode, the DNA which is there, they are going to separate. Why? Because our DNA is acidic. It is also negative. Negative repels. Positive, the opposites which are there, they are going to attract. The ones which are similar, they are going to not attract. They're going to repel. So over here, repellation happens, which is exactly why the DNA separates. They move towards the positive side, okay? They move to the positive side. There is one very important question that is being asked. Which one moves first, the larger fragments or the smaller fragments? What is your answer? Which one is going to move first, the larger fragments or the smaller fragments? In gel electrophoresis, which one? Small one, the small one. Yes, that is how we lose another marks. Listen to me carefully. We do not know which one separates first. We do not know which one separates first. Any of them can separate first. If at all you are asked this question, which one moves faster through the gel electrophoresis chamber, it will be the smaller ones. Please make sure you know that. Okay? Please make sure you know that. So both of them, it can be large or the small one which can move out first, but always remember the smaller one will be the one to move. They move faster. They move faster. Next question that I have for you guys. Okay, next question that I have for you guys. So the first question that I said, the answer is not defined. Why? Because large or small, anyone can move first. If I asked which one moves faster or farther, obviously it will be the smaller one that moves faster as well as farther. Next thing that is there, DNA will be moving on the surface of the gel or through the gel. On the surface of the gel or through the gel. What do you think is the answer for this particular question? Any idea? Through the gel. Exactly, SVV Mata. It is through the cell, and this is known as sieving effect. Why not you said that? It is known as sieving effect. Over here, what do you have, dear, to show the sieving effect? You have agarose gel, which you take from the seaweed and things like that. I'm sorry. Okay, so it shows the sieving effect. Basically, the DNA is moving within the gel out there. Now, suppose over here, I want to remove this particular DNA. I want to take out this DNA for my own purposes. What is that particular process of removal of the DNA from here? Any idea? Of removal of DNA from the chamber or gel electrophoresis, what do we use? Ma'am, you didn't talk about spooling. Spooling is the same method that chilled ethanol. You get the DNA and then you use spooling. Yes. What was my question? I forgot my question. Where is Philip Johns? Listen, I forgot the question. This is my problem. Can you explain tissue culture? Yes, I'll be doing that, dear. Okay. What question did I ask? Okay, yeah, the question was, how do you remove the DNA from here? Blotting. No, this process of removal of DNA is known as elution. It is known as elution. When you take it out, this is the term elution. Okay, the question was, how do you remove the DNA from this particular gel out there? This process is known as elution. Works? Yes, perfect. Now, how do you view it? Before removing it, obviously you have to view it, then only you can remove it. So, as I said, you use the stain that is ethidium bromide. Ethidium bromide will be giving what? A bright orangish red color under UV light. Under UV light, it will give bright orangish red color. Done? Yes, that's all. And that's how we are done with it. Is it clear to everyone? Yes. Finally, listen to me about one more thing. Please know all the steps which are involved there. I can only help you with this. One more thing is there, bioreactor, no diagram, you just go through it, all the important things. Bioreactors, why do we need it? For basically mixing everything really well, and also for making sure the oxygen is available throughout. That is why we have the thing in between. It is known as stirrer. Stirrer makes sure that everything is being mixed well, especially oxygen is being mixed well. That is why we have a stirrer in the stir tank bioreactor. Okay, apart from that, what is downstream processing? Downstream processing mainly relies on separation and purification. Okay, separating things from everything else, whatever is needed. We want to just improve what we just need the protein, so we take it out, and then we need to purify it also. These two things are the major thing that comes under downstream processing. Apart from that, obviously you need to do the quality checking, you need to add preservatives, all of it also comes. For the first two things is separation and purification. Okay, yes. Ma'am, NCRT is enough. NCRT is enough. Yes, more than enough. Okay, but at this particular point, obviously, if you just rely on your NCRT to understand things, it will be difficult. It will be a huge process. Most important diagrams. Who is asking me that? The first video out there in the channel, the first video out there right now is the most important diagrams. Go watch it. I put so much efforts, you know, to make the diagram. Watch it. Okay. Finally, going ahead with the next thing. Ma'am, please, electronics and hardware. Sadly, dear, I do not have anyone over here who probably might be a pro at that particular subject. If at all there was, would have definitely helped you out. Okay? Ma'am, we have been told just to study the model papers. You have to study the model papers, but please go ahead and please go ahead, study extra things also. Ma'am, break. Not now. Okay, guys. Fine, let's go on to the next chapter that is there. 53rd question. I hope this chapter is clear. For everyone, do you have any questions related to biotechnology, principle and processes? Shall I go ahead with the next chapter? Sanjana, everyone is asking for break. Guys, when did we start this lecture? At 3 o'clock. It's been 1.5 hours since I'm talking. I didn't realize it. I can give you 5 minutes break. Will that work? 5 minutes breaks work, guys. We need to cover it up, da. Some people, no ma'am, as in for break, or you do not want a break? Okay. Okay, fine. 5 minutes break, okay? 5 minutes Kit Kat break. What is the time now? 4:32. So we will come by. Okay, we will come take break until 4:40. Okay. Be back by 4. And when you come back with a lot of your friends who are finding difficulty in biotechnology and application. If at all you listen to me, all the doubts will be sorted. Trust me on that. Yes. Ma'am, what's my mother tongue? My mother tongue is Malayalam. Okay. Okay, come back. hello. Hello, Sanjana. Guys, are we back after the break? You know what? Your teacher is such an epic person. She scolded you with the mics off. So you might have seen me going, doing all those things. Actually, I was scolding. I was saying that, where are all the children? So previous year, there were like 300, 400 people out there. And now this year, have no one take biology, or what is it? I'll be taking ecology. Don't worry, dear. Okay. So I told everyone that when you come back, bring hundreds of children with you. So please come back with hundreds of children. Everyone, come on, go share it with your friends. Now we're starting off one very important thing, jokes apart, and that is biotechnology and its application. Okay. So I hope we are ready to begin. Give me a thumbs up if at all everything is clear. And yes, please share it with your friends. Come on. Ma'am, please tell me what, dear? We are equal to 100, ma'am. Ma'am, I don't have 100 friends. I do not know what to tell you guys. Okay, so you saw the answer. I saw that. But yes, still try. Statement number 1. The plants genetically identical to the original plants are called soma clones. Is it true? Statement number 2. Soma clones can be obtained through micropropagation. What do you think is the answer for this? What do you think is the answer for this? I'll move aside. Ma'am, give me an important question. Dear, I have given you, go to the channel and see. Short questions. Listen, I cannot give you the entire paper. No, PU board will arrest me. And neither do I have that. So I have given you the most probable questions out there. And in yesterday's live also, we did a lot of things. Make sure you watch that. Or if you just want the questions, it's there in the channel with fixed question tag. Okay? Ma'am, please tell about soma clones. Acha, I have gave you the question, you gave me the answer. Okay. Seema says the answer for this question is D. Ma'am, all my friends are CS. It's okay, dear. Okay, C is what? Both statement 1 and 2 are true. Is that correct? Listen, the plants genetically identical to the original plants. For example, over here, I had a plant. Okay, suppose I had a plant. Now, what did I do? Jasmine, okay, Jasmine went ahead and cut this particular portion of the plant. Why did she do that? Because this is known as the apical meristem. This particular portion that you see is known as the apical meristem. What do you mean by that? Basically, this region has so many meristematic cells. The cells which have the ability to divide, and divide. So I took a part of it. Okay. Now what do I do? Basically, it is now not a part of the plant, which is why I'll call it as X plant. Just like the X out there, right? X. So it is a X part of the plant, so it is known as X plant. Now what did you do? Basically, you put it inside a media. You put it inside a media such that this particular part, my dear children, will grow and keep on dividing until it forms a bunch of cells out there, basically a tissue out there. You particularly have a name for it. What do you call this? You call this as callus. You call this as callus. Okay. Now what do you do with the callus? Basically, you transfer the callus in another medium that will be having rooting hormones, that will be having shooting hormones, and that is how you're going to get many plants out there. Now you tell me, I took this from this plant. Don't you think the baby plants which are going to be formed over here, all of them are going to be similar? Or do you think it is going to be different? It is going to be similar. No, Baba, we have taken the same cells out there and the same cells are being used right over here. This is known as tissue culture. First of all, this is known as tissue culture because I'm culturing the tissues out there. And over here, if at all with the help of tissue culture, I am basically making almost thousands of plants out there. This will be particularly known as what? Micropropagation. What do I call this? Micropropagation. Why? Because I'm making multiple plants out there with the help of tissue culture. Now the plants which are being formed over here, the plants like every plant which is there, they all look similar, Baba. They are known as clones. They are known as clones. So what do you have specifically? What do you call these things as? Soma clones. You call this as soma clones. This is super important. Okay, understood? Fine. So here the plants genetically identical to the original plants are known as soma clones. Yes, exactly correct. Soma clones can be obtained through micropropagation. Yes, micropropagation is something that comes under tissue culture. So you have got the answer. Okay, ma'am, is tomorrow's bio paper easy or hard? Bhavishwani. Hard. What will you do? If it is hard, what will you do? You'll stop studying? No, right? I always tell this, forget about it being hard or easy, just give your best. Okay, just give your best. We'll see. If you didn't study, it will be hard for you. If you study, it will be easy for you. Think like that. Okay? Think like that. Always. There's no question. What if I say easy? You will not study, you know. If I say it is easy, what is going to happen? You will not have that vigor to go ahead and study for it and probably do something. What if the paper comes to be tough then? What will you do? JJ ma'am, you told that paper will be easy. Now it is hard, right? So yes, I am on the safer side. Okay? Go ahead and give your best, guys. You still have time. Fine. Now coming to this particular question, that is, there is a term that is known as somatic hybrids. You might have heard about soma clones and also somatic hybrids. What do you do with that? What is the meaning of that? How are they different? The difference that comes between soma clones and somatic hybrids is very, very simple. Listen to me carefully. Somatic hybrids are basically for example, I have potato cell and I have tomato cells. You know, in the case of plants, they have cell wall. In the case of plants, Baba, they have cell wall. Suppose I am removing the cell wall over here. The cells which look like this, which not have the cell wall, I will call this as protoplast. What do I call this? Protoplast. And then what do? I am going to fuse this protoplast together. What do I get over here? Hybrid. Potato, tomato, pomato is what I get. This is what? Hybrids. And these type of hybrids which are there are known as somatic hybrids. So always remember, soma clones means clones. They look similar. Hybrids means two different variety of things are being joined over here. Got it? This is known as somatic hybridization, the process, and that is totally different from tissue culture. Is this clear? Everyone, give me a thumbs up. If it's clear, just give me a thumbs up. BT and its application, everything will be covered over here, okay? If you prepare, yes dear, that's what most of us are doing because we do not have a choice. Yeah, you can go according to the blueprint. Blueprint as in the blueprint released by the government, right? Yes, perfect. Now, identify the correct statement. Okay, identify the correct statement, guys. Bacillus thuringiensis produces an insecticidal protein. Oh, okay, let me see that question. Just a second. I think for understanding those questions now, we need to come to this topic. So we'll keep this question on hold. This is 54th question. I'll make you understand what is Bt toxin and stuff like that, and we'll come back again. Okay, so we'll deal with a longer question first. So my dear children, according to your model question paper, this year in MQP, transgenic animals was being asked, genetically modified plants was being asked, and biotechnology in the field of medicine was being asked. So in three model question paper, three different things were being asked. That means we cannot deduce what is going to come over here, right? Can we deduce? No. So what do? We studied everything that is there in the NCERT and we studied in this particular lecture very fastly. Okay, done. So biopiracy, RNA interference, all those things can also be coming in. The steps, the questions related to all this thing, we are going to deal with that. So I will deal first with the Bt cotton and RNA interference, and then we'll go with the topics that is there in the MQP, because in MQP things have been discussed so many times, right? So let's go first with the Bt cotton thing. Okay. One of the applications of biotechnology is to get pest-resistant plant. Justify the statement with reference to Bt cotton. How with the help of Bt cotton can you get pest-resistant plant? So listen to me, I am going to tell the story of Bt cotton. So what is there? There is a bacteria. Bacteria's name is Bacillus thuringiensis, which is why we call it as Bt. This particular bacteria which is there, it is super happy because it has a gene. What gene? The cry gene. It is actually another psycho out there. It makes the insects cry. How? Basically, listen to me carefully. Over here, suppose this particular bacteria is there on a plant. Suppose this bacteria is there on a plant. Now, I am a caterpillar. I come and eat the plant. This particular caterpillar comes and eats up this particular plant. Basically, what's happened? It is also taking in the bacteria inside. Listen to me carefully. The cry gene over here is going to code for a protein. It is coding for a protein. This protein is basically nothing but a toxic substance. It is a toxic substance, but it is inactive. But it is inactive over here in the bacteria's body. You will call this inactive toxin as prototoxin. Prototoxin. Is that clear? You will call this as prototoxin. Now you might ask, why is it inactive over here in the bacteria body? Because of one big reason, that is bacteria's body does not have the pH. Okay, the pH in which probably the prototoxin can be converted into active toxin. It is inactive. You want to convert it into active toxin. Okay, so where will that particular ambience be created? Inside the caterpillar, or basically inside certain group of insects. Why? When the insects take it up, my dear children, and when it reaches the mid-gut region, you know, in the mid-gut region of the insect, the pH is alkaline. The pH is what? Alkaline, as in basic. The prototoxin which is over here suddenly gets the alkaline pH and basically converts into the active form of toxin. Now what happens? This active form of toxin is going to cleave open over here. It is going to cleave open over here, and all the things are going to be released and the organism literally dies. That is the story. Always remember, the prototoxin is nothing but a protein, but it is inactive in the bacteria body. Why? Because alkaline pH is not there. But here it is active. Why? Because here in the midgut region of the organism, my dear children, alkaline pH is there, and basically it is going to target the epithelial cells which are lining the midgut region and slowly creates perforation and kills the whole organism out there. Is this particular thing clear? If yes, just give me a thumbs up. Okay, just give me a thumbs up. Monish, ma'am, I'm JA student who took biology. I do not know anything much. Is it possible for me to get 45 in my theory? Yes, dear, please go to the WhatsApp. Okay, please join the WhatsApp channel. You go to the description of the video. Go to the WhatsApp channel. I have sent a separate message at the end. Read it out. There you find all the links that you need to follow. Go. It will help you. Ma'am, once more. Okay, fine, done. Once more. Again, didn't understand. Okay, guys, listen to me carefully. It's simple. Bacteria is there. Bacteria has its own genetic material, right? There is a gene whose name is cry gene. The cry gene is going to code for a protein. Genes code for proteins, right? This protein is what? An inactive toxin. You call this as prototoxin, the inactive form of toxin. Fine. That is the whole scenario that is there in the bacteria. Bacteria is carrying this particular toxin out there, but it is inactive. Okay. The bacteria is there on a leaf. A caterpillar came and ate the leaf. Along with it, it ate the bacteria also. Now bacteria, suppose I am the bacteria. I am inside the gut of the caterpillar. What happens inside the gut of the caterpillar? Here it is alkaline pH. So my prototoxin, the inactive toxin which is there, it becomes active. When it becomes active, what is going to happen? It is going to target all the epithelial cells of the midgut region, and then it is going to damage them, leading to perforations such that the caterpillar where I am in dies. You get it? Did you understand it now? Okay, can you write in our sentence? Yes, you can. Did you understand it now? Everyone? Jia, Riddhi, and Chammu? Yes, perfect. Okay, this is the thing. Now listen to me very carefully. There's an extended thing about cry gene that you need to remember, and that goes like this. It goes like this. So there are multiple types of cry gene, okay, and I really need you to remember it for sure. There is cry 1AC and there is cry 2AB. Okay, these are specific for killing cotton bollworms. Cotton bollworms. Cool. Okay, now what do you have, Baba? You have another one that is cry 1AB, that is specifically going to kill corn borers. Now, do you think I will leave you like that and I'll ask you to remember it as such? No. There's a way that you can use for remembering it. This is only a trial and error method. You can obviously do it by your own end. Okay, I'm just here trying to help you out. So the thing is that first letter is C, that is cotton C. Second letter is B, that is bollworm. So cry 1AC first and cry 2AB second. That is what I meant. Now you might feel that corn borers also have C and B, but cotton bollworms is a bigger name. That is why you need 2 genes. You can remember like that. You do not need actually 2 genes. Basically, these 2 genes are going to code for proteins. That is the thing that is out there. Okay, either of them will be there. That is there for killing. Next thing that is there, cry 1AB for corn borers. Sometimes we think it is cry 1AC or 1AB. So 1AC is at the top. You know for what reason I told you. Okay. Now, how do you remember cry 1AB? Only one cry gene is there for corn borers since it is a small word. Okay. First letter is C and the last letter is B. C and B. Okay, is this clear now? Will you be able to remember this? This is important, comes in MCQs. This comes in big questions, this comes in MCQs. Okay, RNA interference. Yes, dear, I'm going ahead with the next one only. Perfect. Okay. So please take a screenshot. It will not there in the PDF because it's an extra thing that I've added up. If you want to take a PDF, say if you want to take a screenshot. Okay, we'll read out the question as well. So it goes like this. Some strains of Bacillus thuringiensis produce protein crystals, okay, protein crystals, insecticidal proteins that kill the insects, lepidopterans, coleopterans and dipterans. Please remember, they are all different classes of insects. Lepidopterans, coleopterans and also what? Dipterans. Now, protein crystals form during certain phase of their growth. Okay, the Cry gene, Cry1AC and Cry2AB from thuringiensis coding for the toxins are transferred to cotton plants. Proteins coded by these genes control the cotton bollworms out there. Bt cotton plants express the Bt toxin gene by producing toxin that provides insect resistance. What is there? Anyways, Bt toxin exists in inactive prototoxin form. Insects injects the inactive prototoxin, converts to active toxin at alkaline pH of the gut, which solubilize the crystals. So basically, in the alkaline pH, what is happening? The crystalline thing which was there, the protein crystals which were there, they are getting solubilized. That is how it is going to show it's working. Now, toxin binds to the surface of the midgut epithelial cells, create pores, and that is how it's going to create the death also. Zainab, ma'am, really want to meet you in person once. Oh, that's so sweet. Come to DVLC once. Okay, come to DVLC once, we'll meet. Okay? Fine. Let's go ahead. Okay, so next question that we have is, what is the RNA interference? How does RNA interference help to develop resistance tobacco plant for what do you call nematode infection? Okay, so let's go ahead. So over here, with that, I remember some people I see in metro, okay, and I literally become so happy. Some recognize me, some do not. But yeah, so whenever I see a student sort of people, I really get excited seeing that, oh my God, probably they might be, watching the channel or things like that. I think like that, okay. But anyways, yeah. So over here, what do we have? RNA interference, yes. Okay, perfect. Okay, so let's go. So what do we have over here for RNA interference is that, Baba, listen, this is tobacco plant. Okay, suppose this is a tobacco plant and these are the roots of the tobacco plant. And here comes our next guy who is there, whose name is what? Meloidigoin incognitia. Melodica sab bhul jao, right? Meloidigoin incognitia. Please know the spelling of it. Okay. Now, this guy is basically a nematode. Nematode means they come under Aschelminthes. This guy gets to the root of the plant and then destroys the plant altogether. It causes crown gall disease and things like that. It's a very big trouble in tobacco plant. Okay, how does it do that? Basically, it is able to make its own protein out there. You know, just like any other organism, it will be having its genetic material, that is DNA. DNA is going to make mRNA, right? And the mRNA undergoes translation to form the proteins. And these proteins made by this guy is going to affect the roots of the plant. Now, there is only one way in which I can inhibit it. There is only one way in which I can inhibit it. How can I do that? With the help of interference. What am I interfering over here? The mRNA, you know, it has to be single-stranded. For what? For the process of translation to happen. Then only the ribosome will come and attach and do the process of translation, right? What if I am making this single thing mingle? What is going to happen? It is not going to remain single anymore. It will be double-stranded in nature. Do you think it will be able to undergo translation? Do you think it will be able to undergo translation to form proteins? First tell me yes or no. Big fan, ma'am. Good. Thank you so much, ma'am. I love you. Thank you, Sanjana. Thank you. Why not you? Okay. Yes. No. Someone is saying. Riddhi says no. Sunita says yes. Will it be able to make the what do you call proteins? No, Baba. If at all you have a double-stranded mRNA over here, it is not going to undergo translation. It will not be able to make its protein. Right? It will not be able to make its protein. I forgot my question, by the way, but I think that was the question. So that is our main motto. We do not want to make the protein up here. That is the main motto. And for that, we have to make it what? Double-stranded. We need to make the mRNA double-stranded. That is the only target that is out there. How do we do that? We take some genes from here, from the same guy itself. Okay, we take some gene over here from the same guy, and what are we going to do? These genes we are going to put inside the plant. We are going to put inside the plant. How can we put inside the plant? Now, what is a vector that you use for transferring the gene inside a plant? Gene of interest inside a plant. If you want to transfer a gene inside a plant, which vector do you use for that? Which vector do you use for that? 7:30, dear 7:30, I'll be taking what do you say? What was that? MCQs. I'll be taking MCQs from the question bank directly. So I have made some PDF out there from the question bank. From PDF, we'll do fast. Okay. Someone was asking institute. I'll give the answer for that. Okay, just a second. Plasmid. Which plasmid, guys? Come on, I'm asking you a question, guys. If at all you want to transfer the gene of interest into a plant, which vector do you use? Everyone, I taught you. Come on. Think. If you forgot, think. Think for a second. Which vector do you use for plant? We know vectorless methods, okay, microinjection, biolistics and stuff like that. In the case of plant, what do you have as a vector? If you need a vector. I will not go ahead until you give me the answer. Someone gave me the answer. Jennifer, Agrobacterium tumefaciens. Exactly. Okay. Mansi also 80. Agrobacterium tumefaciens. Guys, remember Agrobacterium tumefaciens, TI plasmid. We remove the transfer DNA. We put the gene of interest and then we transfer it to the plant. For animals, we have disarmed retrovirus. Please remember that, no, I didn't ask about the enzyme that you need. Okay, I asked about what vector do you use. You just have 2 vectors, 3 vectors altogether, plasmid, Agrobacterium tumefaciens plasmid, and then you have retrovirus, right? So Agrobacterium tumefaciens is something that you're going to use for the transfer. Here also, you're going to put the gene into the TI plasmid of the Agrobacterium tumefaciens. This Agrobacterium tumefaciens infects the plant. That is how we are going to transfer it. Okay, that is how we are going to transfer it now into the plant. What happens inside the plant now? You have the gene. What does this gene code for? This gene was taken from this guy. We put it inside Agrobacterium tumefaciens because we cannot directly inject it. So this guy now, here we put the gene of interest. It transferred over here. The plant has the gene now. What does the gene code here? Okay, what does the gene code here? The gene is going to code for a double-stranded RNA. It codes for double-stranded RNA. That is the gene coding for. Now, what is the use of double-stranded RNA over here? Listen to me. This double-stranded RNA is of this guy only, right? Now, this guy is making mRNA inside the plants, and the mRNA is about to undergo translation to form the proteins. This is going to happen. What if this double-stranded RNA which is there, it comes over here and one strand of this RNA binds over here? What is going to happen? The single guy mRNA turns mingle, right? Turns mingle. Now it is double-stranded. Do you think it will undergo translation? It is not going to undergo translation. Ma'am, but it is double-stranded. No, only one strand came. What happened to the other strand? The other strand will get degenerated later on. It will get degenerated later on. Okay, listen to me carefully. I am repeating again. I know that it might be a bit tough for remembering everything all together. Okay, but listen to me carefully. First thing that we have is Meloidigan incognitia. We learned the basic of it. That is, basically, it will be having its own DNA. The DNA will transcribe into mRNA. The mRNA will translate into proteins. And these proteins are something that is toxic for the plant. What if we make the mRNA double-stranded? Will it be undergoing translation? No. And that is our purpose over here. That is our main target over here. We want to make it double-stranded such that translation does not happen at all. Okay, for that, what do? We take the gene from this particular organism. Okay, we put it inside the Agrobacterium first. Why Agrobacterium? Because you need a vector to transmit it. You cannot directly put it inside the plant. So with the help of the Agrobacterium tumefaciens, it has the TIA plasmid. In the TIA plasmid, I am going to put my gene of interest. Now the Agrobacterium tumefaciens goes and infects the plant, but while it infects, it is only passing the gene of interest. Okay, it passed the gene of interest. What does it do? It codes for double-stranded RNA. This gene is coding for dsRNA or double-stranded RNA. So you have 2 strands of RNA being formed over here, right? Now, when you come back to this particular scenario, there was single-stranded mRNA. It was going to undergo translation. What happens is that since this RNA also belongs to this guy only because we took the gene from that guy, it will be complementary to the mRNA. So what happens? The strand which is there comes over here. One strand, suppose this is the first strand and this is the second strand. One strand will be binding with the mRNA. Makes what? Complementary thing, such that it is double-stranded now and it will not undergo translation to form the proteins. Got it? Aren't we interfering the RNA? That is why it is known as RNA interference. If you still didn't get it, ask me once more. I do not mind it until and unless you understand this. Ma'am, QB and MQP is enough. No dear, you have to go ahead. Yeah, QB and MQP is enough. Yes. I thought you were just asking about MQP. QB and MQP should be enough. Jennifer, please, once again. Definitely, I can do that. Okay. Fine. So, what do is I remove all this thing, because I know it can create confusion. There is a plant, this is what? Tobacco plant. There is a root of this plant where an organism is infecting, Meloidigoin incognitia. How does it infect? Basically, it produces some proteins which causes some disease over here. Now, the normal way to make a protein is what? Here from this organism, it will be having its DNA. The DNA will be basically making an mRNA, and this mRNA is going to undergo translation to form what? To form the protein out there. And as I said, this protein which is there, it is toxic for the plant. It is toxic for the plant. Now, I want to stop this process. I do not want to harm the plant. Okay, I want to stop this process. What do? I will have to stop the process of translation. How can I stop the process of translation? The mRNA which is single-stranded over here, okay, single-stranded over here, I am going to create a complementary strand for this mRNA. Then what will happen? It will become double-stranded. If it is double-stranded, do you think it will undergo translation, Baba? No, it will not going to undergo translation. So this is my target. I want to make the mRNA double-stranded. This is what? This is our target. Now, how do we do that? If you want to make it double-stranded, basically you should find a complementary strand to it. You will get all this information from the same guy itself. You will get the information from the same guy itself. So what are you going to do? Basically, you are going to take a gene from this particular guy. This gene is something that is going to code for a double-stranded RNA. This one will be coding for a separate, I'm not talking about this double-stranded RNA, it will be coding for a separate double-stranded RNA. But I cannot put this gene inside the plant directly. Okay, why? Because it's a plant, I do not have any other technique. I know that is biolistic method, this and that, but I'm not going to use that. I need a vector over here. Who is the vector? Usually the vector that we have is Agrobacterium tumefaciens. Agrobacterium tumefaciens. You know, this particular vector has a plasmid. It is known as tumor-inducing plasmid. I remove the part that causes the tumor and basically I put my gene of interest. The gene that I took from Meloidigoin incognitia. Okay, I am going to put it over here. Now, the Agrobacterium tumefaciens comes, infects the plant. It is going to come and infect the plant, and when it infects the plant, what is it passing? It is passing the plasmid along with what? Along with the gene of interest. It passed the plasmid along with the gene of interest. Yes, it did. Okay, definitely it did. Now what happens? Now over here in the plant's body, the gene is there. The gene is going to form the double-stranded. It is going to code for what? The double-stranded RNA. Does it code for double-stranded RNA? Yes, it does. It does. Fine. It's clear until here. Is it clear until here, everyone? Is it clear? Yes, perfect. Okay, going ahead. Now we have got the double-stranded RNA. What do you do now? This double-stranded RNA is going to go to the mRNA. This mRNA was single-stranded, right? I will make the diagram of mRNA right over here. So this is our single mRNA which is there. It is single-stranded. It is about to undergo translation to form the protein. But we make a stop over here. How? Because this double-stranded RNA which is there, it is going to come over. It's there in the plant. This mRNA is also there in the plant. The organism put it out there, right? So this double-stranded RNA came. This is the first strand. This is the second strand. They get separated. One strand comes over here. So this guy moves out of this and comes and binds over here, creating what? A double strand with the mRNA of the organism. Now, if it is forming double-stranded structure, do you think it will be able to undergo translation now? It is not going to undergo translation. It will not be able to make the protein. So what are you doing? You are interfering over here. Okay, you are interfering over here. You got it? Very, very, very important. Yes. Desai, I hope you understand that this is not the place where to find love, okay? See you really soon. Bye-bye. Okay, now. I like your way for teaching. You do not do it for views. Thank you so much, dear. Okay, so I hope it's done and dusted. Yeah, dear, you can watch that, and also if at all you can watch this also. Perfect. Cool. This is RNA interference. If at all you want to take a screenshot of this, please take. The most usual question that comes is, what is something that causes the interference? It is single-stranded RNA, because here only one strand comes and joins, right? Or it is double-stranded RNA. It is actually double-stranded RNA that comes and joins. So the actual guy is this guy. It's very, very important. Double-stranded RNA denoted by dsRNA. Okay, how do you write the answer? What is RNA interference? How does RNA interference help to develop resistant and tobacco-infected plants? It involves silencing of a specific mRNA due to complementary double-stranded RNA. It involves silencing of a specific mRNA due to complementary double-stranded RNA, okay? Molecule that binds it and prevents translation of the mRNA. Identifying the nematode gene. First thing that you have to do is identify the nematode gene. Scientists first identify the genes in the nematode that are essential for its survival or ability to infect the plant. Now what do you do? Using Agrobacterium tumefaciens, we deliver the silencing gene to the tobacco plants. With the help of the Agrobacterium tumefaciens, we are going to deliver it. First of all, the identified nematode gene is inserted into the genome of the vector. Vector is what? Agrobacterium tumefaciens. Then this DNA sequence is then inserted into the plant's genome. Right? Agrobacterium infects the plant, and now this particular gene is now in the plant's genome. Okay, now what? That's all. Finally, production of dsRNA in the plant. Once the DNA sequence is inserted, the plant cell begins producing double-stranded RNA, dsRNA. Now what? RNA interference in nematode. This dsRNA triggers RNA interference in the nematode, silencing the target gene in the nematode cell. Or basically, you can say that one strand of it comes and joins with the mRNA, and that is how it is going to go. Now, as a result, the nematode can no longer function properly and can cause harm to the plant. This is the basic thing. Now, I know this is elaborated. You can write it in your own language. Children, now we are living in a technological era, right? And if we feel that I am not able to frame my own answer, give it to ChatGPT. Okay, give it to ChatGPT and write, I want the easiest way to write this answer. It will be giving you something like that. Rupashri? Yes, they will be. But please underline what's important. RNA interference, double-stranded RNA, Agrobacterium tumefaciens, all these things are important. Myeloid and incognitia, all of it. Okay? Works? Perfect. Guys, now we'll come to one question out there, which was this. Traditional breeding techniques have failed to keep pace with demand and to provide sufficiently fast and efficient systems for crop improvement. In this context, explain how the tissue culture technique has contributed to the crop improvement program by mentioning any 5 of its application. Now this is something that is confusing. Listen to me very carefully. Crop improvement program is there in sexual reproduction and flowering plants, but there they mean the production of hybrid crops as an artificial hybridization. Here you are talking about what? Basically tissue culture, and basically to increase the production of the plants. That is what the concern is. So what do you do if at all you want to increase the production of the plants? You will do tissue culture. Please know that. So depending on the question, your answer should be changing. Now you have been clearly asked, it's clearly mentioned for you. Sometimes it is not. That is why I am telling you. Right? Now in tissue culture, what's the use of tissue culture? Regeneration of the whole plants from the explants due to property of totipotency. First thing, the first best part of the story is that basically I can regenerate a whole new plant from only some part of the plant, right? Why? Because the some part of the plant that I am taking up over here, meristematic cells, they have totipotency. Totipotency means that ability to become anything that they want. They have the ability to form whatever cells, a whole new plant as in. Okay, that is the first good point. Second point, rapid production of a large number of plants in a short time through micropropagation. So in tissue culture, I told you, if at all you are producing so many organisms out there, what do you call this as? Micropropagation. So if at all you want a production of large number of organisms, you can take the use of tissue culture, or basically, specifically, what do you call this as? Micropropagation. Now the third thing, production of genetically identical plants. For example, I find a plant that is pest resistant, that is this, that is that, is super good. Okay, now I want to make genetic copies of these plants. What can I do? I just need to do tissue culture. Okay, so I can get soma clones also. Now the fourth point that is there, commercial scale production of crops like tomato, banana, apple, etc. So these things which might take a lot of your time, probably they can be made literally very well, easily. So commercial production will also be increased. Final point which is there is super important. Recovery of virus-free plants by culturing the meristem. So you know tissue culture is the best way to produce plants. Why? Because over here you can get virus-free plants. Suppose I am a plant and I am infected with virus. I am infected with virus. Always understand, even if I am infected with virus, some parts of my body will not be infected with virus if I am a plant. And these parts are known as apical meristem, axillary meristem. So apical meristem is basically the tip of the plant. Suppose over here. Similarly, axillary meristem is the place from where the branches or probably a new leaf comes up. You call it axillary and then apical. So always remember, apical and axillary meristematic cells are going to be virus-free. They will not have the virus, which is why I can take the plant and produce what? Virus-free plants as well. Cool. So these are few very important things. Sometimes altogether in a mind it does not comes up. So please know these are the points that you have to write. First thing, regeneration of a whole plant is possible. Why? Because of totipotency. Secondly, you have large number of production being through micropropagation. Third, you can produce genetically identical plants for the plants which are really good, and they are known as soma clones. You can scale up your commercial production, and also you can get virus-free plants. And the things that you take is what? Apical and axillary meristem. This is very, very important. They can ask you which part of the plant do you take for virus-free production. Clear? If it's clear, just give me a thumbs up, everyone. Yes, perfect. Thank you. Okay. Now we come to the next question, that is, ELISA is one of the methods of molecular diagnostics. What is the principle of this technique? Name the disease which can be detected by this particular method. So please remember, ELISA is the usual thing that we say is there for HIV virus detection and things like that. But in your NCERT, in the same chapter, one line is written about the, what do you call this thing? That is polymerase chain reaction. Do you know that particular thing? So I'll read out the line for you guys, if at all. If at all, what if you get a question with that? Biotechnology and its application, PDF. Just give me a second. Okay. Yes. PCR is now routinely used to detect HIV in suspected AIDS patient. It is being used to detect mutations in the genes in suspected cancer patients too. It is powerful technique to identify many other genetic disorders. So in your NCERT, yes, I know ELISA is for HIV AIDS detection, but PCR is also now used for HIV AIDS detection. So please know that as well. Okay, for HIV AIDS detection and also for finding any mutations in the genes in the case of cancer patient, PCR, remember. Okay, ELISA is a major thing, but this is also there. This is given on page number 183. Okay. Ma'am, when will 7 o'clock live end? It's not 7 o'clock, it's 7:30 by the way. Okay, it's 7:30, not 7 o'clock. I have to make the PPT, not a PPT, PDF, and it ends by 9:30. Okay? 7:30 to 9:30. Whatever can be completed, we can complete that only. I'll not stress because you also have to study your thing alone, right? Yes. So whatever we can, we'll complete by that particular time. Fine? Yes. Okay. Now, ELISA is one of the methods of molecular diagnosis. What is the principle of this technique? That is a question. Principle of this technique is antigen-antibody interaction. That means suppose over here, I already have antibodies which are there, which are AIDS specific, as in I have seen that people who have AIDS produce these antibodies. It is known to me. So I have kept the antibodies ready. Now suppose I have a human being over here who is concerned that whether they have HIV. What do? I take their blood. In their blood, my dear children, what is going to be there? If at all they are infected with HIV, they will have the pathogen. I'll call this as the antigen. They will have the antigen, right? Now, when I put the blood over here, basically when I treated it with it, I get to an understanding that over here, the antigen is going to come and bind to this location. Only when it is of AIDS, right? Because these antibodies are specifically for AIDS, right? For HIV viruses are there. That is why they will come and attach. But how will I be able to see that? Again, I'll need a secondary antibody. I will have a whole different antibody out there. So a whole different antibody comes over here. It attaches, okay, and basically this particular antibody is attached to an enzyme also. You call the secondary antibody, it is attached to an enzyme as well. Now over here, I am going to add a coloring substrate. When I add the coloring substrate, the enzyme will act on it and give me a color change. It will be giving me a color change. If it gives me color change, that means that there is an antigen-antibody interaction that has taken up right over here. If there is no color change, that means there is no possibility of that person to have, what do you say, AIDS out there. So all of it is basically, all of it is basically linked to what? The antigen-antibody interaction. That is the principle of enzyme-linked immunosorbent assay. That is why you call it as enzyme linked. Why? Because it is enzyme that is being linked over here. So first thing, a specific antigen or specific antibodies attached to a plate. So there can be ulta also. Okay, sometimes they can put the antigen over here and they'll take the blood. In the blood, if at all there is HIV AIDS, obviously antibodies will be produced against it. So those antibodies can be collected. So in the similar way it can be done. It can be either this or that. Okay. Now, fine. I hope you got it. So patient sample is added. If the patient has the matching antibody or antigen, they will bind together. An enzyme-linked antibody is then added. Coloring agent is also mixed. The enzyme reacts with the chemical to produce a colored substance. Color change means the antigen antibody is present. No color change means you are safe. Okay, now name the disease which is detected. Obviously HIV/AIDS. Obviously it is what? HIV/AIDS. I'm telling you again, PCR also can be used for detecting HIV/AIDS, but the primary one is always ELISA. PCR is used to find three things. If at all you just have little amount of pathogen, you use PCR to find that little amount of pathogen. Second thing, HIV/AIDS. The third thing, if at all you have any mutations in the gene, for example, cancer patients will have, for finding that also you have PCR. Three things, remember that, you'll get marks. Okay, guys, listen to me very carefully. For those of you who cannot attempt the live, that is the next live that is there is 7:30, it's okay. You need to have your priorities being set. I tell you as a teacher, I always have told you this thing. Keep this clear in your head. If you feel that I have to prepare for myself and I cannot attend a live at this particular point of time, you know it better than me. You know yourself better than me. You know how much is left. If you think that so much is left, I have to revise them, that is my priority as of now, do that. You do not need to attend the live. Why I am taking the live out there? There might be some children who probably might have studied everything, who might want to do just the MCQ questions, and I am not solving all the chapters MCQ question. I cannot do it within 2 hours, you know that, right? So obviously I'll be taking some chapters MCQ questions only. And that is why I said that 7:30 to 9:30 I'll be finishing up. But if you think you do not have time, please do not attend the live. You go and study for yourself. That is your work. Okay, you know that better. Please, and I will not be sad if at all you're not there. Trust me on that. I will be literally happy if you score good by doing, the best you can. Okay, so that's all. One of the best live classes of you. Thank you. Thank you so much, Vinay. Okay, perfect. Let's go ahead with the next thing. Explain the application of biotechnology in the field of medicine. MQP1 question number 40. So this is a biotechnology in the field of medicine. There are two things. There is insulin production and there is what? Gene therapy. These are the two things which is there. So first we'll go to insulin therapy. Okay, insulin therapy, production of insulin. What is the use of biotechnology in the field of medicine? Two uses are there. First is insulin production, where what is happening, my dear children? You know how insulin is being formed? There is A peptide and there is B peptide, and both of them basically are linked together with a C peptide. This particular structure of insulin is known as the prohormone. Why do you call it prohormone? Because it is not the mature form of hormone. This is not the working form of hormone out there. Now what do you do? Okay, what do you do to make it mature? Basically, you need to remove the C peptide out there. You will be removing the C peptide. You have A and you have B, and over here you will be having disulfide bonds over here. Now you will call this what? You will call this as mature form of the hormone out there. Remember, it's a hormone. Okay, it's a hormone. Now some hormones can be proteinaceous hormones and things like that, but over here I am telling you, remember it just like a hormone, nothing else. Cool? Yes? Yes. Now, how do we make it? That is the question. So initially, insulin is there for what? Insulin basically does one very important thing. You know, in our blood, there is so much amount of glucose molecules. There will be so much amount of glucose molecules. These glucose which is there, it has to be trapped and it has to be converted to what? Glycogen. Otherwise, in our blood, the level of glucose goes off high and we will be having diabetes. So to make sure it is not there, what do? We convert. So the insulin which is there, it converts the glucose to glycogen, right? That is a major function out there. In that way, we prevent diabetes. Now, in some people, the pancreas might not be working well, which is why insulin production might not be happening smoothly. In this particular case, obviously, you need to take insulin from outside source. So usually cattle and pigs and everything were being used for transferring the insulin outside. Okay, but some people started developing allergies to that, which is when Eli Lilly came into the picture. Eli Lilly is a company. Okay, Eli Lilly is a company which came into the picture and said that, why not make human insulin itself? Why are we taking insulin from other sources? Why not we make human insulin? But how can we do that? With the help of E. coli. With the help of E. coli. So what they think is that, okay, let's take E. coli separately. In one E. coli, we'll put one chain. That is the chain for A. Or probably, there is a question, first question that should be coming to your mind is this. What if we put the whole gene for insulin inside the E. coli? What is going to happen? Do you think it will be forming the insulin, the actual, mature form of insulin? No. Why? Because they do not have the enzymes to cut the C-peptide chain, which is why we'll not get the mature form of insulin. That is the problem. It's a E. coli. It's a prokaryote, not an eukaryote, right? It does not have all the enzymes needed. So obviously that is not possible. We cannot insert the whole gene. So Eli Lilly said that, why not put the gene for A peptide in one, put the gene for B peptide in another? Why not make them separately and later fuse them in the laboratory condition? Why not do that? And that was being done, and that is how you got the human insulin, which is known as humulin. That is how you got the humulin. Is this particular thing clear? Guys, what is happening? Why there is mesial retraction and things like that? Is this clear to everyone out there? If yes, let me know. Give me a thumbs up. Hello, Shrikant. All 2, 3, 5 from your all videos is enough for 5 for all your, ma'am, practicing all 2, 3, 5 from all your videos is enough for 60 plus, right? Hopefully it should be. The extra questions, Ria, you were there yesterday, you solved it with me together, right? So yes, it should be working. Apart from that, the previous videos also is there, so yeah. Okay, guys, done. Thank you for telling me it's clear. Now we go ahead. This is the first function of biotechnology in the field of medicine. Now the next thing that we have is gene therapy. Next thing that we have is what? Gene therapy. Now gene therapy is what? Basically, listen to me carefully, everyone who is there. This is one. Okay, this one is super important. Listen to me carefully. Okay, over here what is going to happen is that, suppose there is a person over here who basically has cells, but within the cell they do not have the gene for adenosine deaminase. Adenosine deaminase. You know, this is a very important enzyme. What does this enzyme do? Basically, it does not allow the pileup of any toxic substances. It breaks down all the toxic substances which is out there, right? So that is adenosine deaminase. Over here, this person does not have this gene. Why? Because it got deleted. There's a deletion of the gene that is happening over here. Okay, now what do we do? In their cells, obviously there will be toxic buildup. What can we do then? We do not have any choice. We need to do something, right? So people came up with a thing. They said that, why not let's do gene therapy. What is gene therapy? And where is it being performed? So listen to me. In a normal cell, if at all there's a toxic buildup, it's still okay. But in a cell like our lymphocyte, the one which is a primary line of immunity, lymphocytes, no, B cells, T cells, if they are being affected, that is a very big problem. So for them, we need to do something. Otherwise, our immune system is going to go so low. So what did we do is that we took the lymphocytes from here. So first one is isolation of the lymphocytes. Okay, we did that. First step. Second thing which we usually miss is culturing of the lymphocytes. So all the lymphocytes which were there over here, they were cultured in the medium. They were cultured in the medium. Now what do we do? Basically, with the help of a vector, we are going to put this particular gene inside all of the cell. Inside all of this cell, I am going to transfer the gene. So transfer of the gene is being done. Gene of what? Adenosine deaminase gene is being transferred. Now when I say you need a vector, guys, you tell me, for animals, what are the vectors that you use? For animals, what are the vectors that you use? Come on. Retrovirus. Exactly, Sujay. Exactly, Mansi. Now you got the understanding. Micro-injection, that is a vector-less method, dear. Okay, you're talking about vector. No, you're talking about vector. Lysozyme, everything, they're enzymes. Vectors are only two, Agrobacterium as well as this. So here, in the case of animals, we need retrovirus, disarmed retrovirus. So basically, with the help of disarmed retrovirus, I'm writing also. Disarmed retrovirus, we basically transfer the gene into the cells. Okay, how do we transfer it? Basically, we transfer the cDNA. What is cDNA? Any idea? cDNA is also known as the complementary DNA. Basically, it means that, you know, DNA which is there, it forms the hnRNA first. Okay, hnRNA later undergoes splicing, because here you have introns, here you have exons. You remove, the same thing happens over here, introns and exons will be there. You remove the introns, you just have the exons, and that is what you call as the mRNA. So this is what we actually need, just the exons, right? So here we do not put the entire DNA inside, rather from this mRNA we do reverse transcription such that we get only the coding sequences, and that is known as complementary DNA, cDNA. So we put the cDNA inside this. I'm just telling you for your understanding, it's not there. Okay, it's not there. Shrikant, yes, please stop spamming. Okay, yes, perfect. Okay, so over here I put the cDNA. What is going to happen to the cDNA? It will basically produce the adenosine gene. Now transfer of gene has been done. Now what do? We transfuse it back to the patient. So we transfer it back to the patient. Now what happens? Do you think it's a permanent cure? Obviously no. Why it is not the permanent cure? Why it is not the permanent cure? Just a second, guys. What? Srikant, tell me. What is your question, dear? Is there a question that you want to ask? Yes, tell me. Go ahead. If it's in my capacity, definitely I'll answer you. Okay. PDF notes would be found in the WhatsApp channel. Yes, and you can join the WhatsApp channel. The link is there in the description section. Fine, done. Now listen to me. I'm telling you, Shrikant, dear, if you have any doubts, wait for a concept to be done. I'll take up your doubts at that particular time. You can ask me the doubt. Okay? Important questions are also given to you. Everything in the WhatsApp channel, if you join, you'll get everything out there. Done. Cool? Yes. Come back. So, yes, is this a permanent cure? Answer is no, it is not a permanent cure. Why? Because the lymphocytes which you have just put in, they will be dying after some time. They have a limited period of time. They are not immortal. Now, this is gene therapy, and gene therapy can be done in embryonic stage also. Understand? It can be done in embryonic stage. For example, when we are in embryo, at that particular time, if I do gene therapy, that will be a permanent cure. So, definitely there is a permanent cure, but it has to be done during the embryonic stage itself, and I think someone told also. Yes, yes. And one more very important thing which is there, that is bone marrow, bone marrow treatment can be done where from someone else you can transfer it and things like that. Or probably you can do enzyme replacement therapy where you repeatedly giving this enzyme, but again you have to do it again and again. So bone marrow transfer, or probably the enzyme replacement therapy, or a normal gene therapy, all of them, they are not permanent cure. The only permanent cure is gene therapy done during embryonic stage. Okay? Yes? Works? Okay, perfect. So now we go ahead. I hope, children, it is clear, and over here I have given you the answer properly. Go through it, read through it. Please remember, it's pro hormone. Do not write pro things and stuff like that and come in. Okay? Khushi, good. Hello. Good afternoon. Good evening, actually. Okay, children, clear. Yes, Manju, I can. Please give me that question. I will be solving it. Okay, I do not have that question over here right now. Perfect. Okay, so if you want an exact question. Okay, now we have one more question from your MQP1, and that is this, that is genetically modified plants have some good things. What are the good things about genetically modified plants? So I'll finish it off fast. Listen to me carefully. These plants, my dear children, they can be sustaining with limited amount of nutrients or probably minerals. So we say they have efficient mineral usage. They have efficient mineral usage since they are genetically modified. Second thing, they are resistant to pest and diseases. For example, you have Bt cotton, you have RNA interference method. With the help of this genetically modified plants, my dear children, they are basically resistant to all this particular thing. Next thing, there will be reduced use of chemical pesticides. Why? Because we are transferring the gene directly, genetically modifying it. So do we need chemical pesticides? No. And when we are not using chemical pesticides, obviously the soil quality, the plant quality, automatically it becomes good. Then we have what? Tolerance to abiotic stress. Okay, tolerance to abiotic stress basically means that there are some plants which probably in normal condition might not be able to survive when too much amount of salt is there in the soil and things like that. But genetically modified plants, my dear children, they can survive. Okay, they will be able to survive. Why? Because they have the genes, they have the resistance and everything. So tolerance to abiotic stress. Next thing is improved nutritional quality. I hope you guys know that this is one very important thing. There was golden rice, and the people of Africa, they were fighting, they had malnutrition, lot of things, they had big problems, and that's exactly when, that's how I remember, that's exactly when we have this golden rice being made. Golden rice is nothing but the rice which is infused with vitamin A. Basically, they have the genes for production of vitamin A, that is why they look golden in color. And with the help of vitamin A, your eyesight and everything goes on really well. So with the help of genetically modified plants, yes, obviously you can increase the nutritional quality. Example for that is golden rice, which basically has vitamin A. And the last point that you have is basically reduces post-harvest losses. Post-harvest losses basically means that After the harvest also, there are chances that bugs and some things might come in and destroy the whole thing. But in this particular case, they might be resistant to that also. Post-harvest losses will also be less. So these are the few things that is there. Okay, works? Okay. Going ahead, which we have briefly explained the uses of transgenic animals. If it is clear, guys, give me a percentage. Give me the thumbs up. Yes, ma'am. Percentage of adenine is 20 in doubles, and find the percentage of guanine, thymine, and cytosine. Yes. Just doing this question just after this, okay? Last question. Okay, perfect. Don't worry, guys. Spammers will come, spammers will go also, because we have the tools for letting the spammers go. Okay? Yes, perfect. Application of transgenic animals. What do you mean by transgenic animals? Basically, they are the ones which have genetically modified. In the case of animals, we call them transgenic animals. Now listen to me. The most famous transgenic animals that we have is what? Mice. Okay, in most of the cases, it is mice. Now, what is the use of it? First one, to study the normal physiology and development. For example, I have a gene. That gene is coding for insulin-like growth factor. I want to know the function of insulin-like growth factor in my body. Okay, but I cannot put it in my body because I am a human being. That is how it usually goes. So what do you do is that basically you put it inside the mice, which is similar to us, very much similar to us. Okay, not in looks, but in general genetics. When you put it there, you get to understanding what function does it have. Okay, does it have an effect on growth? Does it have an effect on development? Does it have an effect on any other body functions out there? That is the thing that is there. Okay, so study of normal physiology and development. This is the first point that you have to write, and the example for that is insulin-like growth factor. Okay, yes. Ma'am, do we get a live for computer? I don't think so, because ma'am might be busy. No, I don't think so, dear. Okay, next thing, disease study and human models. For example, I want to know how can I treat cancer. How should we treat cystic fibrosis or arthritis or Alzheimer's and things like that. What do we do? Basically, again, there might be some cases where the same scenario is being repeated in a mice's body, and now we study what is happening, what are the things that are being affected, or probably I can create drugs and see, does it have an effect on these cells, does it kill the normal cells, is it all okay out there, and all these things. Okay, so it is created as models for human disease study. Please remember the name of diseases: C square and A square. Cancer, cystic fibrosis, and arthritis and Alzheimer's. C square and A square. Next thing, biological products, for production of products, my dear children. For example, again, there is a protein, a very, very, very important protein, not a hormone, a protein, alpha-1 antitrypsin. It is being produced inside our body, and it is very useful for the treatment of emphysema. Emphysema is a respiratory disorder. For treating it, we need alpha-1 antitrypsin. Can I tell my body to produce it? Obviously no. I cannot tell my body to produce it. So what has to be done over here? Only one condition. I can take the gene and I can put it in an organism and make it produce it. So production of biological products, I can use this. So please remember, alpha-1 antitrypsin, that is for emphysema treatment. One more thing is there was... Guys, what are you saying? D. Okay, you are solving some other question, is that? Is everything fine? Guys, which question are you solving? Am I missing anything? Ma'am, in the chat. Okay, fine. Listen, come back. Okay, fine. Okay, there is a transgenic cow out there. What does a cow do? Cow basically, what does a cow do? Anyways, listen to me. So there is a human milk protein that is only found in humans, and that is why it is known as human milk protein, alpha-lactalbumin. It is known as what? Alpha-lactalbumin. What are we doing? We are basically making a cow produce it. Now, the cow is producing nutritionally rich milk, and that is insane, right? Well, this is a transgenic cow, and please remember, it is not alpha-1 lactalbumin, is just alpha-lactalbumin, and this is alpha-1 antitrypsin. Super important. Now, the next thing is vaccine safety testing. For example, I want to test whether this vaccine is safe or not. First of all, it's done on the organisms out there, and then on us, and then on us. Next thing, chemical and toxicity testing. Chemical and toxicity testing basically means that over here, I want to check whether this much amount of chemical will be acting like a drug or not. So what do? We make the organism sensitive and then we give the drug. Then we see what is happening up there. If at all the sensitive organism is giving huge problems out there, that means that probably we have to lessen the toxicity out there. So that is toxicity testing as well. So these are the things that you need to keep in mind. Is this question clear to everyone out there? By the way, who was solving other questions, guys, were you listening to me? Is it clear? Is it clear? Hello, Ritika. Ma'am, for common bacterial disease, do you have to write the causes? Cause, yeah, every disease, please know the cause. Perfect. Okay. Perfect. So, now over here, we have one more question. Give reasons for the following. Even though Bt toxin can kill many insects, it will not kill the bacteria that produces it. Why? Because it does not have the alkaline pH out there. Even though Bt toxin is inactive, it kills the insect which enters its midgut. Why? Probably because it provides the alkaline pH. The gene encoding cry protein inserted into the crop plant. Why do you put the cry protein into a crop plant? To make it pest resistant, basically resistant to lepidopterans, dipterans, coleopterans. That's all. Okay, now come listen to this part. Okay, guys, you saw the question. Anyways, we were stuck at this question, remember? Bacillus thuringiensis produces an insecticidal protein that can kill Meloidigoin incognitia. It produces an insecticidal protein that can kill Meloidigoin incognitia. Is it correct or wrong? Guys, give me the answer for this particular question. Guys, what do you think is the answer for this particular question? Definitely. Human reproduction chapter will give you a 5-mark question. Diagram based. Mansi says C. Gangadeep says C. Okay. Sunita bro. Okay. Mansi says C. Kidzone says D. Okay. Let me see. Masha Ali Khan is also there. Identify the correct statement is being asked. Bacillus thuringiensis produces an insecticidal protein that can kill Meloidigoin incognitia. Wrong. It kills insects, not nematodes. The cry gene, cry1AC, can control corn borers. Wrong. It is what? Cry1AB. Cry1AB. That kills corn borers. Now, using Agrobacterium vectors, nematode-specific genes were introduced into the host plants. Yes. Bt toxin exists as protein and converts into protoxin inside the insect gut. Wrong. It is there as protoxin and later it will be converting. Right? Mohinder, hello dear. Okay, perfect. So your answer for this question is going to be C. Now coming to two questions. No. One more question is there. Wait. Where is that list? Yeah. Guys, this is the last question and then I'll solve someone's question. Someone asked me a question. You have to tell me the name of the thing, okay? Detection of pathogen using antigen-antibody interaction. What do you call this as? Antigen-antibody interaction. Detection of pathogen using antigen-antibody interaction. What is this particular process? Come on guys, fast. ELISA, exactly. It is ELISA where you use antigen-antibody interaction. Perfect. Okay, the Deepti. Sunita, also Mansi, perfect Kid Zone, everyone. Identification of mutation in a gene, which is the process with the help of which you do identification of mutation in a gene. Ouch. Manjunath, I'm coming there, okay? Last, I'm so sorry, dear. Identification of mutation of a gene. Guys, we studied this. Identification of mutation in a gene, I told you. B, answer. Everyone write the answer is A, B, C, D. For B, I'm asking. For B, what is the answer? PCR, exactly Sujay. I told you PCR has 3 functions. Apart from ELISA, PCR is also used for HIV understanding, okay? Second thing, it helps in understanding even if little amount of pathogen is there. Third thing, if mutation is there in a gene, okay, in cancer patients and all, for that also use PCR. Okay, so remember PCR, Sujay, Kidzone, Kuvelesh and Sunita, everyone. Masha Ali Khan, Mansi, perfect. Okay. I will be taking KCET after this, dear, okay, after the exam. Detection of the pathogen before the development of disease symptoms. Before the development of disease symptoms, if at all you are going to basically detect the pathogen, what do you think is the answer for that particular question? Any idea? Okay, guys, just a second. The probe, yes. Even I was confused. Guys. There is a chain. Just a second, okay? A single-stranded DNA tagged with radioactive molecule is allowed to hybridize as complementary. Clone having mutated gene will not appear on the footage because the probe will not have complementarity with the mutated gene. Alternative identity. Okay. So the thing is, it's kind of confusing because, okay, listen to me carefully. There's a quick change, okay? Quick change. So in your NCERT, I'll read the line and I'll listen to me carefully. So for PCR, it is said, it's now routinely used to detect HIV in suspected AIDS patient. It is being used to detect mutations in gene in suspected cancer patients too. It is now a powerful technique to identify many other genetic disorders, and also it is used to find very low concentration of bacteria. This is for PCR. So mutation of gene is mentioned over here, but they're specifying that is for cancer. Now for probe, as you guys were saying, okay, a single-stranded DNA or RNA tagged with radioactive molecule. Okay, there is a strand of DNA or RNA. It is tagged with a radioactive molecule, and it is allowed to hybridize with the complementary strand. Okay, it will go and bind to the complementary strand. It will only bind it if at all it's similar to this, right? Yes. Now you understand the logic. If this is mutated, do you think it will go and bind? No. So, which is why it is written that the because of this, probe will not have complementarity with the mutated gene. So yes, here also the probe can be used. So for mutated gene, it can be probe. You are correct. So since I think, correction, okay, since I think over here they have not mentioned mutations in a gene of cancer, we can go with probe as the answer. But if at all they are mentioning cancer, then you should be going with PCR. This is actually a wrong question. They should not be putting a question like this. Anyways. Okay, it's kind of very confusing. Okay, anyways. So, children, apologies. Make it probe and make it ELISA. ELISA though, yes. Okay. Yes, so if cancer has been asked, you can go with PCR. That's not fair though. Okay. Now, next question. Detection of the pathogen before the development of the disease symptoms. What do you think is the answer for that now? Detection of the pathogens before disease symptoms. Any idea? C1. Ma'am, how can I pass? Watch the video that is given to you in the WhatsApp channel. Guys, please do not spam. How many marks do we get from the model for boards? I literally do not have any idea. I'll give you a best shot. That's the only thing that I would say. Kid Zone says it is urine test. Do we do urine test? So over here, again, reading from NCERT. Presence of pathogens such as bacteria, viruses is normally suspected only when the pathogen has produced a disease symptom. By this time, the concentrated pathogen is already very high in the body. However, very low concentration of bacteria or virus can be detected by amplification of their nucleic acid by PCR. Okay, by PCR. Now this is again been mentioned. And now when you guys say urine, okay. Using conventional method of diagnosis like urine analysis, early detection is not possible. So please understand the answer for that particular question is going to be PCR. It is going to be PCR. For early diagnosis, you need PCR. Next thing, control of Meloidigo and Incognitia in the tobacco plant. That is super simple, no? Control of Meloidigo and Incognitia, it is the same thing. That is basically what here you would be having, what do you call, RNA interference. RNA interference. The last part, production of thousands of plants in short duration using tissue culture. Production, BT, who said BT? Arey Baba, control of meloidigoin in Deeksha. Meloidigoin is not, meloidigoin is different. No, BT is different. You don't give me heart attack. Okay? Vijay Lakshmi, I'll take classes of CET after boards have been done. It is not. Soma Glonza, guys, I will not talk to you. Production of thousands of plants in short duration using tissue culture. What do you call this process? I'm not asking you what do you get after the process. What do you call the process? Micropropagation. Exactly, Ankita. It is known as micropropagation. I told you, production of thousands of them is known as micropropagation. The things that you get are known as soma clones. Okay, got it? It is known as micropropagation. Tissue culture, if you go and write that kid zone, you will not get marks. Cool? Yes. Okay. It means identifying the pathogen body before any symptom appear using tests like PCR or ELISA. It's PCR only. Okay, it's PCR. For any less amount of things is there, you find it using PCR only. You do not take that. Ma'am, you're so beautiful. Thank you so much, dear. You're also similarly so beautiful. Okay, now listen to me. Manjunath was asking me a question and also was Riddhi. Guys, come on. Can you pose that question now? Hey, big champion. Is it troll to me? Because I already always call you champions. Manjunath went, Manjunath, I have to... that is not, chat is not available for you. So, Manjunath, you have to ask me that question again. I remember you put something like adenine is 20% or something like that. Right? Adenine is 20%. Then what is the percentage of thymine and cytosine and also guanine? That is a question, right? So over here, what are important drawings? It is there, dear. It is there in YouTube. I have the first video that might be there is of important diagrams. Yes. If a double-stranded DNA has 20% of cytosine, calculate the percent of adenine. That is a question, huh? Okay. Cytosine is 20%. Then how much is adenine, guanine and everything out there, right? That is a question. So it goes like this. Listen to me carefully. Adenine, thymine, they base pair, cytosine and guanine, they base pair. Always remember, take care comes before, okay, in pyrimidines, and Ag comes in purines. Fine. Now when I say 20%, if this is 20%, that means this is also 20%, because how many number of C will be there? That many number of G will be there. Yes, in a double-stranded DNA. Okay, in a double-stranded DNA. Now, basically, what are you doing? Out of 100 is the maximum percentage, right? Out of 100, 20 and 20 has gone over here. 20 plus 20 is what? 40. So 40 is gone. What is left? 60 is left. 60% is left. Now you have to divide that 60 between both of them. You cannot give 60-60. You cannot give 60-60. Basically, you have to divide the 60 between both of them. So you will make it 30 and 30. Got it? Who said 40-40? No 40-40. It is this. I hope you understood. When we say one is 20, other one will also be 20 only. 20 plus 20, 40. Out of 100, 40 is already gone. Now what we are left with? 60. This 60 also we have to divide between them. So 30 plus 30 is 60. 60 plus 20, 80. 80 plus 20, 100. Now it is perfect. Okay? Yes. Cool. You're most welcome, Manjunath. Okay. Now... okay, guys. Ma'am, can we skip ecology? Ecology is a chunnu chapter. No, do it. It's easy. Ecosystem. You meant ecosystem? Okay. Guys, it's been 6 o'clock finally, and now 30 minutes more, and then we'll khatam it. Evolution tree. Yeah, there can be questions. Just go through it. Okay. Say. Now we come to the ecosystem part. So yesterday I was telling you that I want to teach you something, right? And that was this species-area relationship. This was given by Alexander von Humboldt. Please remember, this is from biodiversity. And how about organism and population? Can we skip? What should I tell you? You should not be skipping any of the chapter. Why do you think there is a need for skipping things? If you have no time left, at least there are PYQs. But skipping is not an option because you always need a backup. Now, can you explain the pyramid thingy? Yes, definitely. I'll be explaining the pyramid thingy. Before that, one very important thing, species relationship. How many of you feel it's tough? How many of you feel it's tough? Raise the hands first. Ouch. By the way, guys, I hope you're drinking water. Lycopods, conifers tell important bumps, ma'am. It's easy. If at all you see yesterday's lecture, I think you will be able to remember at least some of it. Yes, so most of them. Okay, let's go ahead, guys. Let's see this particular things. Okay, I'm from Simplified Minds. What I need to do, ma'am? Huh? I didn't get you, dear. Okay, anyways, let's go ahead. Let's talk about this particular guy that is there. So, the logic is pretty much very, very simple. Okay, it goes like this. This is species richness, not diversity. It is species richness. And over here, you have the area. So, suppose you have seen the Dora the Explorer. Dora the Explorer, everyone might have seen any time at the life. Anyways, at least seen the poster. Listen, Dora is walking around. When Dora walks around, she sees a huge number of species. When she moves ahead, she sees more number of species. So what is the understanding that you get? With more explored area, you will be seeing more number of species. But do you think that is endless? It just keeps on going? No. It keeps on going only up to a certain limit. After that, you will not find new species out there. So there is basically one very important thing over here, and that is this particular curve. It says that it increases but only up to what? A certain level. Now, if at all I want to show that, how will I show that? You have studied mathematics, right? And we say that when S is directly proportional to A, it means that however number of species is there, basically it increases with the area that is explored. But we are saying that it goes only up to a certain limit. Beyond that area, you will not be finding anything else. So what do? We basically put a sign over here. It means that, yes, species richness basically is proportional to the area explored, but only up to what? A certain limit. That is what you mean by Z. Okay, that is what you mean by Z. Now, if you remove the proportionality constant, you will get something like this, and that is known as C. Got it? S is equals to CAZ. Got it? Over here, you put that particular thing. This particular diagram is for that. S is equals to CAZ. You might be asked, what is this line signifying? You should be able to answer this. Now, the next thing. If we take the log value, listen, I'm not a pro at this, but I'm just telling you. If we take a log value, you get something like this. The chart comes out to be something like this, and you have to convert into log. How do you convert into log? You say it is now log S is equals to log C is plus what? Log A. Now the Z proportionality which is there, you put it in front. That is a rule of log. Got it? And this particular line is going to signify this particular formula, that is log S is equals to log C plus Z log A. Do not mug it up. That is the worst thing that you can do. I still remember in my grade 12th, I literally mugged it up and I didn't remember. Okay, so please remember. Now over here, what does Z stand for? What does Z stand for? Z is basically the slope of regression. It's basically denoting the slope of regression. And it is very much easy to understand if at all you try to hear me out carefully. Listen, it says that. It says that basically suppose it's something like this. And over here, you have one more line. Okay, you have one more line that goes like this, and there are two slopes. One is for this particular, one is for this particular red line that is there. What do I mean? Here in this particular case where the white line has been mentioned, I am clearly able to see with the area that is explored, the species richness is not too much. Or if we are not able to understand like this, I am going to do something. Just a second. I'll tell what I meant. Okay. First we make a line up over here. Okay, and then for the red one we make another line. Okay, in the white one, what are you seeing? With the area that is increasing, my dear children, the species richness is not too much, but the area is way too much. Species richness is this much only. So the relation between species richness and the area is what? Less. This relation between species richness and the area is less. With more explored area, I am not able to see much amount of species richness. That means we say that the Z value is less. The Z value is going to be less. Okay, that means Z value less means over here whatever has been said is not being followed. We usually said that with more explored area you will see more speciesness, but that is not been entirely followed up over here. Now the same case when you talk about the red line, if area is more, similarly species richness is also more. In this particular case, obviously there is a relationship between both of them, and I'll say here the Z value is going to be more. I'll say the Z value over here is going to be more. Got it? Okay, so when we say Z value is more, that means you will have a steeper thing. This is steep. This is not that much steep. Do you think you can climb this easily or this easily? You can climb this one easily. This is very much steep, my dear children. So always remember, steeper slope means they will have high Z value. Okay, if over here you consider this will be having less Z value. Usually in nature, there are certain Z values which is mentioned in your NCERT. Okay, and that is what? Any idea? Ma'am, little fast. Okay. What is that? Any idea? The Z value which is normally there. Super important question. Guys, I will be fast. Don't scare me. RC. Look, no one is saying the Z value. It's not on 7 o'clock. It's at 7:30. 0.1 to 0.2, exactly. The usual Z value that maintains in the ecosystem is 0.1 to 0.2. This is the usual thing. Do not forget it, do not forget it, do not forget it. But in some cases, obviously things might be different. For example, when you talk about a larger area, in the larger area, the Z value might increase. From what range to what range? Any idea? It goes from, so over here, listen, for very larger area, in most regions, the slope value ranges from 0.1 to 0.2. In very large areas, such as continents, the slope becomes steeper. It becomes 0.6 to 1.2. 0.6 to 1.2, this is only for larger areas. Okay, that means it has high Z value. Now, example for fruit-eating birds and mammals in tropical forest. So when you go to tropical forest, you will be seeing that with more species, more area explored, you will be finding more species of fruit-eating birds and mammals. Okay, so there the Z value is close to 1.15, which is very close to 1.2. That is the maximum Z value out there. You get it? That's all. That's all that I wanted to tell you. Perfect. Is it clear, guys? Is studying MQP enough to score 60 plus? No, you have to study extra. Please make sure you do that. Okay, do that. Do not miss out on your marks. Is this clear, guys? Ma'am, pyramid. Yes. Pyramid is what? Guys, if you have any doubts related to ecology and stuff like that, you can ask me now. Then we might not be available. By the way, one very important thing. I brought this for you guys. You know what is this? This is Parthenium, carrot grass, which is invasive species. For everyone who probably can see. Okay, this is Parthenium, which is known as, where is it? Yes. Okay, you might have seen that. It spreads around everywhere. Okay, now energy pyramid. Which pyramid are you asking, Anik, by the way? So the pyramid looks like this. Pyramid. Okay, if you're talking about biomass pyramid, it is going to be looking like this. Okay, it is going to be looking like this. It is a normal upright pyramid where you have to mention the producer, PC means primary consumer, secondary consumer, tertiary consumer. Please mention dry weight, kilogram, meter, minus 2, raised to the power minus 2. All these units are important. Each of these numbers are important. Please mention producer, trophic level 1. This is trophic level 1. This is trophic level 2, trophic level 3, trophic level 4. Okay, done? Okay. Ma'am, number pyramid is also there. Number pyramid, it looks like this. Where is it? Yes. So number pyramid, usually when you're talking about upright ones, that will be looking like this. The broader thing will be there over here. Producer will be highest. You can give highest number over here. Make the numbers low, 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 low. At the top, the apex predators will be very less. Okay, number of individuals, mention that. Again, trophic level mention what does it all stand for. Give an example of an organism out there. Yes. Okay, energy pyramid, again you have to mention the same thing, but mention joules. Okay, mention in joules. So in your NCERT is in kilojoules or joules? I think is it in joules only. Okay, so please remember, guys, please tell me the exceptions of the pyramids. Where do you find the inverted pyramid of biomass? Where do you find the inverted pyramid of biomass? Just a second. You do not have to remember the exact number and all. Okay, the inverted pyramid. Okay, so energy pyramid is in joules only. Okay, so you do not have to write kilojoules. Again, over here in energy pyramid, one thing that you have to make sure you cannot randomly put any number. Mention, like for example, you can start with 10,000 joules, then 10% law is going to be applied. 10,000, then 1,000, then 100, then 10. So 10% law, it goes according to 10% law, so do not write any number and come out there. It is going to be problematic. Okay? In all three pyramids, which one is very much important? All three are important. Harry, what is there? It's very easy, no? Right? It's easy, it's easy. So pyramid of energy, I'll tell you how to draw it. My God. Children, you guys. Okay. So over here, you write, this is what? The PP. Then you write, this is primary consumer. This is secondary consumer. This is tertiary consumer. Then you write here, what does PP stand for? Primary producer and stuff like that. Here you give the units. How do you mention? Suppose it had 10,000 joules of energy. 10% law applies over here, which is why only 10% is going to go. What is the 10% of 10,000? 10 divided by 100 into 10,000. Do that. 0, 0 cancel. 0, 0 cancel. How many zeros are we left with? 1, 2, 3 zeros. Right? Yes, so basically it will be what? 1,000 joules. Again, 10% of it, 100 joules. 10% of it, 10 joules. Mention all this and please mention this is trophic level 1. Do not write T1 and come, write trophic level 1, trophic level 2 and give an example of the organisms out there. This is how you do that. Yeah? Thank you so much. Okay, now listen, in aquatic ecosystem is where you find inverted pyramid of biomass. Why? Because there usually what do we have? Plants are the biggest one and they have highest biomass in terrestrial ecosystem. In aquatic ecosystem, what is going to happen? Their phytoplanktons are there. They are usually the producers. Phytoplanktons are the producers, right? So what is going to happen? They are going to have very less biomass. The zooplankton which feeds on the phytoplankton, they will have higher biomass. So there you will find the inverted pyramid. It goes like this. Okay, and then the small fishes and then the large fishes. Similarly, pyramid of number also you have inverted pyramid. Where do you see that? Basically, when you talk about just one particular tree. One particular tree, only number of tree is 1, but the organisms which are there feeding on it will be more. So that is inverted pyramid of number. Yeah? Yes, Kovalesh, I will be coming in the morning live and I'll be just taking MQPs, nothing else. Okay? Ritin, go watch the video. Complete biology video is there in the WhatsApp channel. I have put that. Please watch it. Okay. Guys, accurate data for biomass pyramid. Yes, obviously we have to. I mean, just give a normal number. Make it less. That's all. Okay. So these are the few things that is out there. And just give me a second, guys. Is there anything that I'm missing? Okay. So it's not in pond that you generally see. You usually mention it as sea. Okay, it is sea. In ponds there can be plants also, apart from phytoplankton. Yeah, in sea also it will be there. Sorry. Forget. Okay, ma'am, population WhatsApp name. WhatsApp name, dear. Over here, go to this particular thing. That is your description. The first thing which is there. Okay, the first thing which is there. There's a link. Click on that to get lecture notes. You will be finding that. Okay. Now coming back to the topic, someone was asking about growth curve. Yes, Sujay is asking about growth curve. I'll be talking about growth curve, and growth curve goes like this. Very simple. Okay, here you would be having the population density as well as the time. If at all it is going like this, that means there is no end to it. The population is just growing like that. Okay, it is exponential curve. You find what it is known as exponential curve, and this exponential growth is being seen, and the name of the curve is J-shaped curve because it has a J shape. This is naturally not possible. This is naturally not there. But in your question, if at all it is mentioned that ideal conditions are being given, and there is no mention about limiting factors, they are not mentioning anything about limiting factors. Okay, then please understand it is going to be exponential. If they are just saying ideal conditions, or probably they are saying controlled optimum condition. Optimum means everything is nice. Controlled optimum conditions means everything is nice only. But if they are mentioning there is limiting factors, this is going to end sometime and that is going to end sometime, that means you are talking about the next curve, which is known as the S-shaped curve. Why a S-shaped curve? Because my dear children, over here it is not going to grow beyond a level. Why? Because it assumes that every environment over here has a carrying capacity, which is why it will not grow. It will come to a limit. This is the ideal thing which is generally there in your NCERT or anywhere. If you get a question where, okay, if at all you get a question where you are mentioning limiting factors, just understand it is going to be sigmoid curve. Okay, this curve is known as sigmoid curve. Now there are two formulas for this. You have dN by dt, that is population density change, is equals to Rn. R stands for intrinsic rate of natural increase. It is meaning nothing. This means B minus D. Now you have this also. This particular curve will be dN by dt is equals to Rn. Everything remains the same except that you have to mention about the carrying capacity. So K minus N divided by K. By this we mean that there is a carrying capacity. Beyond it, it is not going to grow. Yeah? Yes. Yesterday I solved a question related to it. Go watch that also. Okay? So please do that. And here mention population density, also time. Guys, I had to explain you something important is what I remember. Did I speak about age pyramids yesterday? Did I anyway speak about age pyramids yesterday? Anyone remembers? Age pyramids. Age pyramids, no ma'am. So listen, age pyramids, one more thing, very important thing, is that you have three types of age pyramids, right? One where it is growing, one where it is declining, and one where it is stable. Okay, one where it is growing, one where it is declining, and one where it is stable. And it goes like this. If at all they are saying that the organism in the pre-reproducible age group, so this is pre-reproducible age group, then you have reproducible age group, and then you have post-reproducible age group. In this particular condition, this is pre, this is reproducible, and this is post. That means young people and the ones that are in the middle range and the ones who are old in nature. If you get a curve like this, or basically graph, a pyramid like this or something like this, basically it means that the lower portion is increased, they are in more amount. In future, there are people to basically reproduce, right? So in this particular condition, the population is going to grow. It is going to grow. On the other hand, okay, if we have another curve where it looks like this, pre-reproducible age group as well as reproducible age group, they are in the same level, and post-reproducible is less. We mean that it is going to be stable. Okay, it is going to be what? Stable in nature, the population growth. Now, on the other hand, if at all we have something like this, pre-reproducible is less, reproducible is more, and then basically something like this. Anyway, if at all this is lower, anytime if this portion is lower, that means in future you have very less people to reproduce. It is going to be declining. It is going to be declining in nature. Did you get it? Is calculator allowed, ma'am? Yes, calculator is allowed, dear. Only simple one. Yes. Clear? Apart from it, another question, sacred grooves. I hope everyone knows what is the importance of sacred grooves. We didn't do the questions yesterday, did we? Sacred grooves. Let me see. Did I put any questions on sacred groove? No. Okay. Anyways, do you have an understanding of sacred grooves? Why calculator? I do not know. Someone asked me, I just told. Usually some people, if some terms related to mathematics comes, they might be worried about it. By the way, everyone, have you clicked on the like button? Make sure you do that right away. How to identify autosomal recessive dominant stuff? Listen, autosomal, yeah, liked and subscribed. Thank you so much, Jia. Autosomal dominant disorder is only one thing, and that is myotonic dystrophy. And the easy way of identifying it is that every single generation you will be finding at least one parent to be having the disease. One will be colored. Autosomal recessive means, recessive condition means there will be skipping of generation. Parents might not be having the color change, but their children might have be having. That is recessive. So children usually get confused with sex-linked recessive and autosomal linked recessive. So in sex-linked recessive, it's mostly the males who are affected. So you'll find more of the males being colored. In autosomal, it is usually both of them are being affected. But in your NCRT, for sickle cell anemia, they have given a pedigree for both actually males are more affected. But yes, you do not need to be worried at all. Ma'am, is this all for now? I think yes. Works. Please know about sacred groups and all this thing, biodiversity conservation. Little details are super important. If at all you miss out on that, probably you might be missing out on a lot of things. Yeah. I hope that works. 7:30 there is a class. Yes. By reading 3 model question paper and 2 pre-boards, how much can I score? I literally don't know how much you can score, dear, because there is no guarantee what is going to be coming in. No, diagrams are not for more than 40. Listen, it is going to be definitely 10 or maximum 12 marks. Not beyond that. Yes, you can leave now. So we'll end up this particular live, guys. We'll meet soon at 7:30. I also have to make the PPT and stuff like that. For everyone who wants to solve the questions directly from the question bank with me, be there for the live at 7:30. I'll complete the live by 9:30. We'll only cover the chapters that gives most of the MCQs. Okay? And morning live, I'll be coming at 5:30. Okay, I will be trying my best to come at 5:30, and I'll be giving you a rapid thing for MQPs. We'll try to do as much as possible with the limited time that is there. Okay, perfect. Then see you really soon, and wishing you all the best. Please take care of yourself. If at all we are not meeting anytime soon. Okay, bye bye, dear. Meet you at 7:30. Okay, cool. We'll meet at 7:30 then.